हेलो। हेलो अगेन। से हेलो अगेन। Are you the one? Good morning.
Good morning, everyone. I welcome you all to the 9th DNA Day symposium cum workshop. We will have guest lectures, events, and hands-on workshop for all of you and to all the registered participants. Good morning, everyone. I welcome you all to the 9th DNA Day symposium cum workshop. We will have guest lectures, events, and hands-on workshop for all of you and to all the registered participants. Hello. So we do have a guest lecture right after this. So I would call Shalini to take over.
Good morning. Thank you, Nikita. So I'm from Biomedical Genetics Department. I greatly appreciate this opportunity to be a part of this team organizing this event. So as you're aware, this year's National DNA Day's theme is genes and metabolism, and workshop and guest lectures have been scheduled for 18th and 17th and 18th. So like another exciting thing about this year is that this year is the 200th birth anniversary of Gregor Mendel, the father of genetics. So like once the father of genetics is said, I am convinced that it will not be long before the whole world acknowledges the results of my work. And celebrating DNA Day now, it's commemorating his birth anniversary. It is remarkable because it shows how far we have developed. So like I once again welcome all of you to this exciting opportunity to explore and discover celebrate the 9th National DNA Day. Now I call upon Saujanya to give the introduction to our guest lecturer.
Good morning, everyone. Thank you, Shalini. I welcome you all for the 9th National DNA Day. I am privileged to stand here and welcome you all for the 9th National DNA Day. Today we are here to have one of our most profound delegates, Dr. Badrinath ARS, our beloved senior. He is a medical geneticist and a district consultant of District Medical and Health Office, Andhra Pradesh. He has completed his PhD and postdoctoral fellowship in our very own institute, Vellore Institute of Technology. We welcome you, sir. He has been appointed as scientific officer in the Department of Transplant Immunology and experience of 10 years in the field of molecular analysis of myopia, congenital cataract, and nephrological diseases. He has also been nominated for Indo-UK Scientist Award at Chennai in 2013. I would like to take up this opportunity to call Sir up on stage to enlighten us on the topic, lifestyle or genes, who's to blame? One request, please put your phone on silent or in switch off mode before the guest lecture starts. Thank you.
As per the theme of the conference, it is lifestyle or genes. Usually what is saying genetic matter, related to genetic matter. Or sometimes it is income multiplier. So when we discuss anything about the what we say is that this is fair. That I will be the last, but this is fair is it is difficult, like it is a reason for it is. It can be the environment factor, it can be the daily factor, or it can be the lifestyle factor. So this three individual called as the this is fair. Okay, so this method is like fighting between lifestyle and genetics. Who is responsible for any case? Any case? Any case for lifestyle or the genetics measure only any prognosis? Could we say measure of what? Yes, more. So next question. If we discuss anything related to that person, what is the disease? It is any abnormal condition. Other than normal, it is called abnormal. Other than normal person, any clinical condition exists. We call it as a disease or total disorder or syndrome. Symptoms. Okay. So these are three different clinical categories. So when we say any clinical conditions, the major follow up is like combination of either genital factors or sometimes environment or this combination of these four together with chronic as a multi-factor. So multi-factor means like different factors. You can't say it as a pinpoint factor, but it is a multiple factors. So when we discuss about the factors which affect any clinical condition, it can be the proximal cause, or it can be the distal cause, or sometimes it is a predisposing condition. What we study now is like a predisposition. So any treatment given now, we call it as a, we will have diagnosis in the pre-diabetes. Means we are diagnosing in the earlier stages. So that the treatment will be effective. So these are the major factors in the clinical diagnosis. Apart from that, what we discussed now, genetic environment and lifestyle also plays a important. So as I said, many factors will influence, but who are determinants? We talk genetic, we talk about genetic determinants. What is that? Genetic determinants. Any guess? What is the determinant? Which can be? Okay. So many factors influence the development of ADCs. If we say ADCs, there are many factors like we said, specific or deletion. Okay, many factors. So, investigation of any clinical condition, usually, any investigation of any multifactorial disease comes from the basic clinical observation, like basic physiological or clinical observations. Like first, how it is happened, if any disease, diabetes, for example, of cancer, how it happened. It can be environmental or it can be genetic. For example, if we say environmental, the person is predisposed or it has a predisposition to any clinical condition. But if the environmental factors pushes, then it will be the stronger clinical, it will have a stronger clinical condition. It means that genetic makeup is already in predisposed condition, but the environmental factors also support this clinical predisposed condition. Then the disease condition will be earlier. Like, say for example, diabetes. Long time we should say about 40 or above 50 but now 30 years, right? The answer is 30 years. So this is one example where environmental factors plays a role. So when you say the risk factors or the risk criteria, we usually say it can be the objective, relative, or attribute. So whatever this, if it is absolute, it is in any of the population. It is absolute is it can be the any of the population. But when it is relative, we are comparing the two different people who are exposed and who are not exposed. Like what we say in experimental condition. Control group and test group. Right. So this is also a relative. So when it comes to attributable, it is nothing but the general observation between the exposure and non-exposure. So whichever is all the three characteristics of the risk factors, what we have taught, all this prior to be placed role. Whatever is the risk factor. So when the conditions are come out strongly, it is genetically predisposed, inherited, like cancers, like breast cancer, certain breast cancers, they are strongly inherited. Other than that, all this multifactorial is major role in any risk factor of the clinical condition. Right? Family history. So when we study the genetics, family history, because family history, what we study family history plays a major role in any of the inherited disorders. But the now clinical conditions other than the inherited disorders, like what is the metabolic disorders, what we are studying now, this also influence family history, also influence the metabolic disorder. Any observation in the 5 years back, what was studied in our BMG lab, that itself is a best example. Under the heading of Professor Radha ma'am, we have studied, ma'am, can I say that? If it, ma'am, can I say that one example of familial inheritance? Diabetes familial inheritance. Yes, ma'am. So this is a research study conducted in our BMG itself. It says that there is a familial inheritance of diabetes. It's not only diabetes, like we say type 1, type 2, right? So in type 2 to type 1 inheritance. So the parents generations are type 2 diabetes. And when it comes to proband or the next generation, they are already with type 1 diabetes. So this is the one of the best example to study why even though metabolic disorders, the familial history will influence. So when we are reviewing a case, like GC people will be there, right? Genetic counselors. So when you are reviewing the case, you should also take this as an important factor. Familial, even though in metabolic disorders, not only in oncology, also in metabolic disorders. So familial history plays an important role in prognosis of any clinical condition. So those dying at younger age have lesser chance of manifesting the disease, or offsprings have lesser family history. This is one case study, like a case study. A person with a smoking habit. Okay. And another person, non-smoker, and he is with cancer, any of the, say, cancer. So what is your observation? Usually what we say is smoking causes cancer, right? Non-smoker, what happened to non-smoker? Why he is getting cancer? Because of? Which can be the triggering agents, right? There are some triggering agents. Any other? The same example, what I say, maybe the family history, right? Family history, but the parents don't have any family history of cancer. But the next generation is with cancer at the earlier stage. What will be the condition? Yes, that can be the one reason. Any other? So we say that exons are the coding and introns are non-coding. Other than that, anything is functional. Anything is functional in the genome.
Other than the exons, is there anything functional component? So, is there anything term called intra and inter? Intragenetic factors, intergenetic factors, what are that? So intra and intergenetic factors also plays a role in familial inheritance, like transcription, epigenetics, what we study, right? Epigenetics are the pushing factors. Epigenetics are the pushing factors. So DNA exons are only the coding regions. Like what we say is they will say what to code, what protein to code. But whenever the DNA is without any polymorphisms or without any SNPs, then the protein will be as usual normal. But whenever these external factors are influencing on a cell or an organ or in the human body or blood system, then the coding will change, which is like what we study is epigenetics. Right? Expression patterns will ultimately change. So along with the familial history, the external factors also plays a role in coding a particular gene. So problematic thinking or disease gene. This slide tells about the relation between the disease and the genes, what I explained now. All the coding happens with the exons along with the contributory factors like expression systems. So only the genes come into the contact when the environment is advantageous or advantageous, like what he says now. If the environmental factors supports for the diseased condition or the predisposed condition, the onset will be earlier. Onset will be earlier of any clinical condition. And ultimately what will happen is lifestyle, healthier lifestyle depends upon the environmental factors and the food what we eat. Our DNA just codes what it is there, but the coding of that or the genetic sequence depends upon the environmental exposures. So if you ask somebody the same question, does genes cause diseases? Genes cause diseases. If you ask a genetics person, he will say yes. But if you ask any nutrition person, he will say the food what you eat depends upon all. How can you justify these two? GC people, any GC people, genetic counselors. That's why it is called, it depends on who you ask. If you ask a general BMG person, he will say because of mutation, it got a particular clinical condition. But if you ask a GC people, they will take a long chart of their observation and they will conclude. Right? So it depends on all what you ask them. But whenever you say a particular clinical condition, ultimately at the end of the chart, what you say is either multifactorial or it can be the combination of familial history and environmental factors. So genetic material instructs proteins, like what I say now, it will instruct proteins and it is a normal process. And there is no any ambiguity in that. And the genes are inherited. Genes are inherited from the generation to generation, what we say. And somatic mutations, the somatic mutations trigger any clinical condition? Does somatic mutations trigger any clinical condition? Yes. What are somatic mutations? Somatic and germline. Somatic and germline. What do you say? Somatic are not germline. Welcome, sir. Somatic are other than germline. other than germline. So other factors are transcriptional factors or cell dysfunction factors, protein synthesis factory. These are all also plays a role in triggering of any clinical condition. Though, so this is the one example. What I'm saying is A and B genetic disease are really normal genetic process unless C and F external factors are involved in that condition. What I am giving an example. So there is a clinical condition. It is a predisposition condition, but if it comes into action along with the external factors only, it will become as a pathogenic condition or any disease condition. So this is an example for that. So what do we study? This all monogenetic effects, all the genetic factors involved in any clinical condition. So monogenetic factors or complex factors, variations in metabolic and environmental toxins and behavior effects. These are all the conditions where we see in the abnormal conditions. So interactions. What are genetic interactions? What are genetic interactions? What are genetic interactions? Answer will be there. BMG. What are genetic interactions?
Interaction between the genetic factors and our lifestyle or environmental factors, when they come together and then they expose or they exhibit a particular trait or phenotype, we can call it as a genetic interaction.
So it is like a triggering factors of or combination of both the environmental and like disease triad. What I said is best example is a disease triad. So this can be the examples. So usually genes are not pathogenic. Genes are not pathogenic. Like what he says now, genes are never pathogenic, but when it has any disruptions in the sequence, protein coding sequence, or sometimes non-protein coding sequence, it will lead to the pathogenic. Usually what scientists say is non-pathogenic is introns are useless or they are not any of use, but they are also directly or indirectly useful in the regular metabolic process. And similarly, no single gene can be attributed to environment because we should have seen the KEGG pathway website. Does anybody know that? KEGG. Okay. So you just go there, you just type the gene of interest, any of your gene of interest. Then you can see how it is interlinked with several related genes. Okay, say, for example, if I say my gene of interest INS, it will have even to mTOR or even to P52. So it has it has interlinked with any many of the even oncogenic genes. So it is a network. In the network, if one gene is disrupted, the surroundings also will get disrupted. So this is one of the clinical factors, like what we study in research, clinical factors. So now, can we have some conclusion on this? It's not lifestyle and genes. First slide, it was lifestyle and genes. Now it is genes versus lifestyle, right? What do you say with that? With all these discussions now, can we conclude something? Which matters for health? Which is important for health? Both are important. Genetic makeup is ultimately important, but at the same time, lifestyle is more important in order to maintain this genetic makeup. So this is what we say. This example, what I say, there are two persons. One is a smoker and another person is a healthier person doing all the regular exercise. But the person who is doing regular exercise is prone to cancer, but not the smoker. Smoker lived up to 90 years. So what do we say for this? GC, genetic makeup, early predispositions, high risk factor, right? This is a combination of all these genetic factors. Then what happens to a smoker? Will he get resistance to that chemicals all he smoke? That's why he lived long. What is in case of smokers? GC people or anybody? Why he is living long? We say that cancer caused by smoking.
And like smoking and like still getting the still not getting the disease. So in that case, we say that the people who was working out and everything, but had a genetic predisposition for that particular disease. But the other person who is a non-smoker, luckily, he does not have the position of that, so he did not do the.
So early, this is a long process. This is a prolonged process. It can't be the, we say that acute and chronic, right? So that is the condition. So if your health is simply written in your genes, what is the point of all that exercise and healthy diet? If your health is simply written in the genes, like what is here, if everything is coded in the genes, then what is the use of taking healthy diet? Why you have to take? Whatever it is, it will maintain the body. Why to take it? Again, healthy diet or lifestyle factors? Why to maintain that? It can be due to, we don't want to give any chance to the in between triggering agents. Like what you say, epigenetics, what we study is outside factors, like outside the cell. So outside the cell should not trigger any of the clinical condition or influence any clinical condition. That is the reason why we maintain lifestyle, healthy diet, etc. That is why genes and studies, genes and health, they are called outliers. Only genes is not responsible, but genes and environment, genes and lifestyle, or genes and diet together always plays a role. Is it? Is it what I said? Is it right? Right? What we studied, disease triad. When we maintain all this disease triad properly, like environment, lifestyle, or genetic factors, we can't do anything with the genetic factors. But when we look after the rest of the two, then only everything will be normal, everything will be healthy. Like if you are also predisposed condition, the clinical condition can be prolonged. Like metabolic disorders, what we say. Metabolic disorders mainly, our theme is itself is a metabolism, on metabolic genes or metabolism, right? Metabolism is half dependent on genetic factors and half dependent on the lifestyle factors. So we have to accept this fact. So when it comes to disease progression is nothing but the time taken by the disease to get into the action, that is the predisposition. Like what I say, metabolic disorder, type 2 diabetes mellitus is a metabolic disorder. What will happen? What will happen? Insulin resistance. Body cannot use the produce insulin. Like whether in type 1, insulin is not at all produced. So type 2 diabetes, what I said earlier is, before years, they used to say 50 years or 40 years. Now, even in 30 years, you are can be with type 2 diabetes. So how we can prolong this? If a diabetic person of 30 years, if we meet any diabetologist or if any GC people like this, what they say is take the diet, healthy diet, take green leafy vegetables. Why so? Why so? It will balance the imbalances. It will balance the imbalances in metabolic process, or say any of the green leafy vegetables will increase the iron, or it will give the get you out of the anemic condition. So these are all imbalances, balancing the imbalances. So this all comes in the disease progression. And cancer, which we say is multifactorial, which is also same multifactorial. This also depends upon the stage of diagnosis. Stage of diagnosis. So with all the lifestyle habits or dietary interventions, we can reverse certain clinical conditions. We can revert certain clinical conditions. So it's not only about lifestyle. Then what I said is also the genetic factors. Staying healthy is mostly a matter of choices what we make. Okay, several environment factors are around us, but how we want to live, what we want to take is depends upon us, right? So our healthier lifestyle makes our health. Coming to epigenetics, research is on only epigenetics now. What is epigenetics? What is epigenetic? Epigenetics. Genetic. Right? Epigenetics is nothing but studying the external factors of a biological component, or a cell. What we say is a cell. So genetic predisposition might not affect you all unless it is triggered. What we say, no, it is already in our genes. No doubt about that. It is the genes are already inherited. If it is any predisposed condition, we have to balance with the healthier lifestyle in some clinical condition, not all clinical conditions, in some clinical conditions. So should we blame this epigenetic changes because they are the external factors responding or transcriptional influencing the transcriptional factors? Should we blame them? Epigenetic factors? Should we blame genetics, or should we blame lifestyle factors? So nobody to blame. Nobody to blame. Okay. All have equal part. All should play equal part. If it is a negative part, then it will trigger the clinical condition. If it is a positive, then it will be the prolonged condition, or sometimes it is a non-pathogenic condition. So this is also a take-home message, like lifestyle will always drive the gene expression. Like means our lifestyle determines the clinical condition, or determines the what to code. Even to the positive or negative. Sometimes it can, like what I say, it can be reversed. Some metabolic disorders can be reversed. So it can be, we can push into the negative part, or we can drag into the positive or negative, whatever may be, vice versa. So this is mostly for the GCs. To what extent is the burden of disease due to different components? What is the burden a patient is having when he sits before you? When a person is attending for a counseling, either pre-operative or post-operative, what is the condition? What is the burden, or what is his mental condition? Psychological stress. Psychological stress. Yes. Yes. If it is a pre-operative or post-operative, will have a physical pain under certain clinical conditions. Any society related? Right? So ultimately, it is a burden for a person who is attending the counseling or the patient, or even though sometimes it is the burden for family members. If it is any pediatric, right? It is a burden for family members. That's why we take into the concept of abortions, right? So these are all a burden of for a group of people. Sometimes it's a group of people, sometimes it is for a person alone. So only thing is to maintain healthy lifestyle, to reverse any condition or to be healthy. This is only the message. So genetics also will play a role, lifestyle also will play a role, and epigenetics also will play a role. They have to play a role if a person has to be in the healthier path. Okay, if a person is said to be healthier without any disease, all the three should play a role. In clinical condition, if anything is known to be diseased, we blame for the disease triad. The same thing will happen if we are healthier. So disease triad has to be taken seriously. So this is the disease triad, what I am saying till now. So any of the environmental factors or the host and the agent which is in the clinical condition, all this will play a role for maintaining healthier lifestyle. So we don't want to blame anyone. So everything has to be maintained or balanced equally. Nothing should be imbalanced. So this is a take home message for all of you. Thank you. So any doubts open for the hall? Any questions?
Hello, sir. I'm Neil Mani, a student of biomedical genetics for masters. Sir, like you already talked about a lots of factors that include environmental genetic factors in the progression of a disease. Sir, in the recent year, like earlier, like a few years ago, whenever we used to talk about like angina or the cardiac diseases, we used to consider age as a major factor. Well, like even in the clinical conditions, well, like even a person is having the heartache or something condition. So if patient is more than 40 years or 50 years, then the doctor used to suspect that, okay, patient can be the victim of angina or the cardiac failure or some kind of thing. But in the recent years, the recent days, we have seen so many incidences where people below the age of 40 or the like on the border age, they are getting the angina or the cardiac failure or the cardiac arrest. So, and in the many of the cases where we have, like it is evident that the people who got the angina or the cardiac arrest, they were following a healthy lifestyle. So what do you think that can be the major factor? Can it be any particular SNP or the genetic predispositions alone, or there any environmental factors which triggered it? Or if even if there is any environmental factors, then what that could be? So because in the certain like small gap of the years, we have seen a quite hike in cardiac diseases, especially angina, sir. So can you please?
So this is only as far as what you said now, this is only mainly of the environmental factors what you see. We all eat pani puri, right? Pani puri. Most of them, I don't think nil. Do you know what is there in pani puri? Long-term complications of that. Do you heard about kala namak? Right? So kala namak is one of the factors for fluorosis. Right? Fluorosis, what will happen? It will eat your skeletal system. Right? Upon prolonged years, you don't have any skeletal support. Right? This is one of the environmental factors what I'm talking. If there is no predisposition in that, genetic predisposition. In the same way, what you are asking is in cardiology, both environmental factors will play a major role. Genetic predisposition is not so much, but environmental factors will play a role. Whatever we eat now, right? You should see what are the contents in that. If you know what you are eating, then it won't happen. If you are eating what you know, it won't happen. That is the major factor in cardiology. Age related is different. But if it is an earlier stage, congenital abnormalities are not the factors all the time.
in many of the cases, we have seen that the victims are either from the sports industry or the popularity business, like the actors or all, like for which they are really good nutritions, and they are like support of good nutritions, lifestyle management and all. And still they are happening. So don't you think like there could be any
Sports injuries are totally different. The case reviews of sports medicine is totally different from this. So regular cases should not be measured with sports medicine.
Do we have any more questions from the audience? So we have few questions from the online participants as well. You can take over the mic, they'll shift. So the first question is, over the years, has the perspective of the lay people towards acceptance of genetic bases and cytogenetics changed? That's the question. So over the years, has the perspective of people towards acceptance of genetic bases and cytogenetics changed? And how does it affect the future of cytogenetics based on the lifestyle?
Cytogenetics is always a gold standard. Whatever we say in genetics, cytogenetics is always a gold standard. Even though what you are going for exome sequencing or next generation sequencing or whatever may be in the next step, cytogenetics are always the gold standard method for certain clinical conditions. Not all, but certain clinical conditions is always a gold standard method. It cannot be neglected, whatever the technology has come now.
And also there's a follow-up. How would you see the light of the difference between Indian mindset and the people abroad? Like, because they are based more on scientific research, more broad, and we have all these Indian home remedies and, people suggesting in India. So how is that?
So when we are comparing different population, like what I say, our hand will have 5 fingers and each are with different height, right? So based upon the availability of the local conditions or local food, it depends upon the area or the country to country, right? We need not compare from one country to another country, but ultimately India is very much advanced. If you say any other terminology for advanced, you can use for that. Even though in thousands of years before, India is always advanced. Only thing is we have to recognize it. It depends upon our students. We have to do that.
Any questions?
So you were in PhD medical genetics and now you are in government.
Yes, ma'am.
So how are you able to manage, because there are many students here, either biotechnologists or biomedical genetics students.
Yes, ma'am.
So what do you think their future is going to be?
So ultimately, our genetic students till now they are placed either at GC or BMG people in industry, R&D or academia. They have widened opportunities. Even in GC people, they are in Cincinnati, abroad in good position, and BMG they are, and GC people are almost in every metros. Wherever you go, there will be one VITN of genetic counselor in any clinical setup. You can cross check with that. At the same time, even BMG students, they are almost all abroad. As far I know, my classmates or my juniors, my seniors, either they are working in medical colleges or they are abroad for research. Genetics is evergreen. We can't say, like what I said, genetics is one of the factors. In this basic point, also, ma'am, we have given a proposal for Andhra Pradesh government to establish a genetic registry. Yes, ma'am. Like we have places with high CKD, chronic kidney diseases. So genetic predisposition is one of the cause for that. So there are certain factors where genetics involves, and government also should take certain steps to have a list with them. So we also given proposal for genetic registry. So even in government side, we'll have. Also, I am the one of best example for that who studied genetics and got into the government. Thank you.
last question, the genetics, like Sir, when we see the trends in genetics, so it started with the like cytogenetics, then comes the PCR, and then comes like nowadays we all are talking all we are talking about NGS and other sequencing. Sir, what do you think the future is going to be? Like what next step? Like we are already done with the cytogenetics, then the PCR, then now the NGS.
Did you ever see chromosomes in microscope?
Yes, sir, I did.
How beautiful they are.
Sorry, sir.
How beautiful they are.
They're pretty beautiful. So they're
So,
really good.
So they are, whatever the years in future, there will be the minimum standards for assessment of clinical conditions. Whatever the technology has come. Sorry, question? That is what your question?
No, sir, my question is like when we talk about the trends in the genetics, right? So earlier, when we talked about the cytogenetics was one of the major standard, and earlier, like we rely on the cytogenetics more. Then there was one more advancement come PCR. Then another advancement is called sequencing and
You are
GS we are
asking the
talking about.
future of
So
diagnosis.
yeah, so now we are talking about like in next diagnostic, what is going to be the next step? It can be the genetic mapping or something others. That I'm trying to ask.
Depends on R&D. What they want, what the clinical demand is. Usually they will go, what is the, how the clinical demand is. Depends on R&D, what they want to take out. Other than NGS.
Thank you, sir.
Thank you. A gentle reminder, a Google form has been sent to all the registered emails. So in case of any questions later on, it will be conveyed through this form.
Thank you, sir. Thank you for taking out your time and giving us this valuable talk. As in today's days, we have noticed that everything we want instantly, we have forgotten how it is important for us to be healthy and to maintain a lifestyle. So today, as we talk, as we heard your lecture, it enlightened us and encouraged us to dig deeper into the research and to know what wrong we are doing. So thank you for your lecture, sir.
Please be seated.
Can we all please be seated as we are going to start the next guest lecture? Today, yeah. Acha.
Hello.
Hello. A very good morning to respected sir, dignitaries, faculty members, and students from all over the country. Today, I shall be introducing our guest speaker, Dr. Gopalakrishna Ramaswamy, CEO and founder of TerraQ's Innovations Private Limited, Bangalore, Karnataka.
Presently, Dr. Gopal Krishna is the founder and CEO of TerraQ's Innovations. TerraQ's has been founded with a vision to develop novel products, assays, and provide leading edge solutions in precision medicine in India and globally. Presently, the company TerraQ's is 5 years old.
Dr. Gopalakrishna has completed his PhD from NCBS with Professor Matthew and UAS with Professor Uday Kumar in the area of molecular biology. Because of his immense interest in RNA biology, he went on to do his postdoctoral research with Professor Furnox Henry at the University of Connecticut Health Center and Professor Frank Slack, who is the co-discoverer of LET7 miRNA at Yale University. He has worked on RNA interference and microRNA mechanism and has contributed in understanding how microRNAs work with proteins to inhibit their translation. He has done his research in a variety of fields and has published his work in internationally reputed journals.
After returning to India in 2005, he worked with the technology company Invitrogen as the first employee and was involved in setting up the company with director Dr. Uday. He also led the team for product development in the area of genomics and microRNA work. To understand the commercial side of the technology business, he moved to Thermo Fisher Scientific and managed the cutting-edge technology portfolios. After that, Dr. Gopal Krishna has worked at Genotypic Technology, a leading genomics company, as general manager and VP, and managed the business for 5 years. He also enhances business skills by completing Executive General Management Program at IIM Bangalore. His varied experience has helped him to view the business and company through multiple views. His area of expertise includes molecular biology, genomics, proteomics, informatics, software business, oncology, and spatial biology.
All these experiences and learnings have helped Sir to run TheraQ successfully for the last 5 years, and the company has become self-reliant and profitable. TheraQ is the first company to get both Encounter Sprint and GeoMX DSP platforms in India and collaborates with pharmaceutical companies and research centers all over India. He and his team are working to develop non-invasive methods or products for monitoring recurrence in multiple cancers. Thank you, sir, for accepting our invitation, and we are looking forward to your lecture. Everyone, please welcome Dr. Gopalakrishna Ramaswamy.
Thank you. Do you guys hear me? I think the last question was kind of a setting up the thing for my talk. I think that's what I could hear. I know a lot of the things. Okay. Yeah. So by the time the technology goes to the actual how it impacts takes a lot of time to look at it. So PCR was probably 1990s, early 90s. It was kind of a discovered, and then now it's everywhere. Then the qPCR, unless the COVID came, probably many people won't even know what is qPCR. Sometimes some of these events help you to hasten the technology or improve the technology in such a way that it impacts the people much better. So what I'm going to talk today is it's a kind of a slightly a digression from what earlier you heard. I know I saw the people who are talking, a lot of them are talking about actual things, what is going on. As a technologist, I look at differently. How do I kind of enable multiple research or multiple diagnostics, or it could be multiple things which how we can enable, because once you put that in the center, you can always use the way you want. So it's important to learn what exactly is available, so that way it enhances the whole thing. So how do I continue? And thanks for Sudesh and then Dr. Saraswati, all the people who are kind of invited me and then to be part of the students. So One thing I accept the talks because I go back and then relook at what I have learned is kind of again anything new has come up or not. So that way it helps me to be on the edge all the time. So before I go to this, I asked a couple of questions. How many chromosome pairs we have? I think everybody will know this. Right? 20, okay. How many? What is your genome size? How many of you know? Right? I thought that should be known by everyone. Okay. How many types of cells we have? Any guess? Some number. Guess it doesn't matter. It could be. Okay. In a way you're correct, but there are, even in the somatic there are multiple types. A neuron cell is different. So how many cells we have in the body? Anybody on an average? Huh? That's approximately that's the number. How many types of cells? How many cells a human being has? 10 to the power of 14. So it's 3.8 into 10 to the power of 14 probably. So that's like 38 trillion cells we have. If you can imagine the bodies every day you get up and then walk, it's a kind of a miracle, isn't it? A lot of us take it for granted. Like many cells and many organs have to come together to do whatever we are doing. So that's an amazing thing. So, but ultimately everything is coded in the DNA, the RNA, the protein, the metabolics. So there are two ways people look at it. I don't know. Many of you might have read the book Molecular Biology of Cell. Anybody? Bruce Alberts and all those things you guys know? Yes or no? Okay. So there are two concepts. One is Alto and I think one who looks at the sky and then asks for all the questions. Another one looks at the earth and then asks for all the answers. One is reductionist approach, another one is the holistic approach. People took it different ways to answer the question. Sometimes both are important. Like you can't just say that I will just take one way to do all the things. So that's where the all these looking at the DNA, what is happening inside that individual gene is causing the disease or not. All these things, sometimes the questions will help you to answer those things. So just to answer certain some of the questions I asked in the beginning, it's around if individuals have around 38 trillion cells, and then around 226 cell types we have. Obviously, everyone answered this question because this is the basic thing everyone teaches. The genome, if you look at it, it's a 3 GB genome we have. It's around 2 pairs. If you look at it, it's into 2 base pairs. And then 20 to 30,000 genes will be there in that. And then that produces around 100,000 plus transcripts. There is always a more number of transcripts are produced compared to the genes. So, and then if you look at it, the proteins around like 500 plus thousands of proteins, like because there are multiple levels of modifications will happen, and then that's how it kind of you are looking at. So that's having said that, you have so many things, but everything to look at it at one shot, it's not so easy. So people devise multiple types of techniques, and then that's how they came to certain individual techniques, and then the multiple techniques for NGS and all those in picture. So ultimately, everything is kind of done by the workhorse is in your cell is protein. Okay, whatever the gene and RNA might be there, but ultimately the protein has to do the function. So many people assays, if you look at it, it's in the proteins. People look at it, and then ELISA and all those things. If they're not able to find out, then they go to the RNA and then DNA. So But again, there are different types of stabilities there. Proteins are very finicky, very difficult to handle. Structure is changed, and then unless you are looking at the liaison or the technique, it's so difficult. RNA is again it's unstable, but at least it's in the strand wise. You can look at it in situ hybridization. All those things you can do easily. DNA is the easiest technique to look at it because DNA any place you can put it forensic after 50 years or thousands of years you can collect the DNA and then look for the changes. So the DNA's stability, if you look at it, DNA is better, RNA is kind of okay, you can still store certain way. Proteins, each day it can change. So people use different techniques to do different things. So depending on what stability is there, technology has developed, and then that's how you can look at it. So if you, just to summarize here, if at all, if you look at the majority of the omics data, if you see it today, people look for the genome sequencing. A lot of the times they look at it, genome or exome sequencing. And then also somebody told about the epigenome. So epigenome is nothing but the modification on the genome, which can kind of ultimately lead to the changes in the protein expression. So that's what is called epigenome. Transcriptome is all the RNAs which we are involved looking at that's what is called transcriptome. And then proteome, people are looking at multiple ways to look for the proteome, and then the metabolome is another thing. I just mentioned the technologies which are kind of being used here. NGS has fueled the technology, like the answering certain omics questions extensively. Whole genome sequencing, exome sequencing, and also RNA sequencing, and also the lot of panel sequencing kind of done by the NGS technologies. Proteomes, majority of the mass spec is used, and the metabolome is kind of HPLC or the mass spec analysis. These are the things that are used regularly. But ultimately, unless you annotate all of them and then put it in a database, it becomes useless. So it's every time you need to recreate certain things. So that's where the database becomes important for all of us to maintain properly. So just to give a kind of a glimpse of what types of RNA might be there, maybe not many people can read this. So that is the way the RNA biology kind of going towards. There are multiple types of RNAs are there. Many of the times in the textbook, there are at least when they are teaching, there are 4 to 5 RNAs they explain. One is mRNA, tRNA, rRNA, miRNA, circular RNA, all this. Even in the miRNA, siRNA, you can have pi RNA. There are multiple types of RNAs are there. So people are interested in its own way to look at how they are connected to the diseases and then how they are developing involved in the development. So this is just to summarize what I showed there. So a lot of the times mRNAs are kind of a protein produced, and then another thing called non-coding RNA. There are 2 types of non-coding RNAs there. One is short, and then another one is a long. Long gives, some will have all poly A, some will not have the poly A. There are multiple ways. In the small RNA, that's where, in fact, in one of the introduction, LET7 miRNA, where I worked with as a postdoc, he was the co-discoverer of LET7 microRNA. That was the second miRNA in 1998, 2000 around. They published in the Nature. So there are multiple types of RNAs. So, but all of them we have to somehow detect and then quantify exactly what is happening inside the cell. Otherwise, it's of no use for just knowing what is it exactly. So just to go back and then ask some questions, like I said, there are only 20 to 30,000 genes are there. How do we get a lot of proteins? Anyone has any question? Answer? Have you heard of who was answering? Yeah, splicing is one. So you know that immunology, if you have read immunology, all these antibodies are kind of produced because of the alternative splice, and also the VDJ, that combination keeps on happening. So if you have 10 genes, you can almost like produce thousands of proteins out of that. So that's how the alternative splicing is very powerful. Just to give a glimpse of, like, for example, if you have a there are, let's say that you have 10 exons. I'm just giving you an example. Out of that, 9 exons can join and make a protein in one cell type. Another 9 can join, make a protein in another. Only 2 exon can join, and as long as it has a starting and then the ending region, it can have a multiple option. So it's the combination of this can lead to a multiple types of proteins. So sometimes if the splicing is not happening properly, you're going to get a genes which are kind of a changed genes, changed RNA if you look at it. That will produce a protein or that will not produce a protein. Ultimately, you end up in a situation where they lead to a disease condition. One of the many of the carcinogenesis genes, either you have a fusion genes or the alternate splicing genes are there. So this is one thing which is very, very important to keep in mind. So another thing, this is called a cis splicing, if you look at it. For example, another 2 genes are expressed separately and then they break because of certain UV radiation. They join together and then they produce a different RNA altogether. That's called the trans splicing or trans thing. So that leads to the fusion genes which are not at all probably present in the situation, because these can join together because of splicing error. It happens, but a 2 different chromosome joining and then happening, that is because of the UV radiation and the exposure of certain environmental factors which Dr. He was mentioning earlier speakers. So the alternative splicing is also helps in the development. So for example, it could be a one type of RNA is produced in your heart cell, another same RNA can kind of entirely different proteins to be produced in the brain. So that leads to the complexity and then also the certain changes. In fact, there are certain receptors and all those things. If you have an alternative splicing, and then if it is not identifying certain molecules, the cells will be always active, especially the EGFR and all those things. It's always active. So that leads to the cancer and other things. So just to give you a glimpse of that, there are splicing and then fusion. They lead to a lot of the changes. In fact, there's a disease called sarcoma, probably many of you might be knowing, or even in the lymphoma, all those things. So majority of the times, the new fusion proteins are produced. People use that as a target to find the new molecules or the target molecules. So drug discovery approaches takes forward. So we know that technologies are needed for the advancing the research. As earlier I mentioned, NGS is one thing. But all of you know that NGS is kind of a technique where you have to extract the RNA or you have to extract the DNA and then you do the sequencing and all this. I'm going to tell a little bit further. There are a couple of technologies which we are working regularly with a company called NanoString. How it is different from the normal technologies, what people regularly do it. So anyhow, this is already she explained about the company. We are in Bangalore, and then these are the 2 machines what we have. This is a kind of a workhorse. This is a smaller one, is a kind of a earlier machine we are using, that called Encounter, and this is called the Geomex. That's a spatial profiler, which I'll explain a little bit later. So any technology, unless if it is a kind of a reliable and then if it is not reproducible, it becomes a challenging thing to diagnose or to screen the assays. So that's where when we were looking at technology, which one to kind of acquire, one of the things which we saw was extremely accurate, and then the high fast, within 2 days the results we will get, and then very easy, and then multiplexing option is there, highly quantitative. These are the things we looked at it, and then we purchased this machine. So just I'll explain how this single molecule counting works. Then I'll go to the Geomex profiler. So what all of you know that there are, for example, if I take a cell or a tissue, if I extract on any given day, there are like 10,000 RNAs, different types of RNAs are produced or synthesized on any given point. But my interest is to look at it pathway. When I'm looking at, I might be interested in 30 genes or 100 genes or 800 genes, something similar to that, or multiple pathways I would like to look. One is if you have RNA-seq, you can always sequence everything, then look for that. But there are some sensitivity issues are there. I'll just come back to that later. But if, and also the clinically they want a quicker result, they can't wait for 4 weeks or 6 weeks to get the results. So what we do in this technology is for each RNA, there will be 2 probes are created. One is called the capture probe, which is a 50 nucleotide complementary to the gene of interest, and then that will contain the biotin. Another one is called reporter probe, which contains another 50 nucleotide complementary to the RNA, same RNA, and then that will have a 4 barcodes, 4 color barcodes, and 6 combination. 4 to the power of 6, like the 4096 possibilities you can have at any given point. So in principle, if I can always create a 4096 barcodes and then look for 4000 RNA in a given RNA solution. So we do on an average around 800 genes at any given point, anywhere between 10 to 800 we regularly run this. So how does it work? We work on it works on the microfluidic method. You take extract the RNA and then keep those complexes, those probes for each RNA. We incubate that in the hybridization overnight and then run on the cartridge. This is the microfluidic cartridge which separates the unbound molecules from the bound molecules. It's like this will contain the streptavidin beads and then it separates, and then it ultimately, when the electrical current is given, all of them, they will come and settle here in the microscopic surface here. Now the machine takes the image of this and then starts counting the barcodes on this. Okay, so this is what the number we get. So it's typically so for example, in each lane there are 192 images are taken, and then those images are counted, and then these barcodes are counted and then added, and then the number is given. That's where the single molecule RNA counting comes. And then if you look at it here, there is no other method called cDNA conversion. Just the RNA I'm just mixing with the probes, and then I'm separating it out and then counting the barcode. That's the only step. So this is how the cartridge looks like. There are 12 samples we can run, and then ultimately you get the information from each lane. You can also do the 96 samples. For each lane, there is a multiplexing option is there. That's called plexet. Each separate barcodes could be looked at into that. So what you get at the end of the day is just a number. So has anyone done the transcriptome analysis here? Like any bioinformatics person is sitting? No. Okay. So for example, RNA-seq, if you do, what is the kind of data you get? Anybody knows? Could be 8 GB, 10 GB data. Usually, typically, because your exon or RNA region is around like 50 MB to 60 MB. Even if you look at it 100 times, if you want to sequence that, it's around 6 to 8 GB data you get. Because what we see here is around 15 KB, 20 KB information. It's an Excel sheet which we get, just the number you get, and then you just do the analysis later. You can almost do the similar analysis similar to the RNA-seq, all the analysis. So it falls in between the qPCR and then the microarray and NGS. So if you look at microarray and NGS, 10,000 plus transcripts I can look at it at any given point. But qPCR, anywhere highest multiplex you can do is 7 to 8 multiplex comfortably. Though people say 12 plex you can do it, but usually 4 to 5 people do, and then maximum 7 plex. Nanostring you can look at around 800 mRNAs, and then you can almost have the same sensitivity as the qPCR. It is in between the microarray and NGS. If you look at the number of transcripts, and then if you are looking for the sensitivity, it is almost similar to the qPCR. So that leads to the since there is no RNA conversion. For example, a lot of them you might be working with certain clinical samples and all those. When you get the clinical samples, majority of the times it's kind of degraded. How much ever you say them, you have to immediately snap freeze, or they will store the cancer samples in the FFP blocks for 7 years, 10 years, and all those things. You have to go back and then relook at what happened to those patients, why they didn't survive after 10 years or after 5 years, why they didn't survive, though the drug was given. So to ask all those questions, we go back and then relook at the FFP section, and then completely degraded RNA you get. This is the RNA which is degraded. This is a bioanalyzer assay. I think many of you might be knowing. In the gel, if you run 18S and 28S rRNA, that's what we see on the gel. That's what is shown here in the bioanalyzer. But in the FFP RNA, it completely gets degraded. What you see is just a 200 nucleated RNA. So that RNA also gives us almost similar to the bioanalyzer 8 to 10 RIN. This FFP sample usually gives 1 or 2 RIN RNA integrating number. It's called a RIN number. So even if it is degraded, you will get the good thing. Why it happens? Because there is no cDNA conversion. Directly, I'm just looking for the RNA hybridization. That's all I'm doing. As the in situ hybridization, I'm doing for 800 mRNAs at any given point. So this is just I summarized why is highly suitable for translation research. Since many of you are in the biomedical and then other things, it will be helpful to know how to use certain really compromised RNA or compromised samples to get the good data out. So I don't know, many of you might be, that is for the RNA. miRNAs, all of you know, no? It's around 21 to 22 nucleotide RNA. It's very difficult to quantify so easily. So what we do is these miRNAs, we add a oligo called bridge oligo. For each miRNA, there will be a bridge oligo is there, and then a miRNA tag sequence will be there. So they both bind to the bridge oligo, and then the ligation site is kind of created. You have a ligase, you add that, then miRNA gets converted into become a larger RNA. It's called an miRNA. I'm extending to become an mRNA in principle. So that's what is called miRNA. That is the way we look at the miRNA quantification. So now when it becomes a bigger RNA, I can just look at and then see what is happening as though similar to the RNA, which I earlier explained. This comprises all the positive and negative controls and also the ligation controls, because ligation is kind of be added as a step here. So these two things we regularly do. In fact, miRNAs are very, we probe on the plasma, we probe on the serum. You must be knowing the urine, all those things contains the exosomal RNA. So liquid biopsy, a lot of you might have heard of the liquid biopsy. They don't want to go and then do the rebiopsy of the tumor of a cancer patient, because once they have taken a primary tissue, if you ask him to, can you give me once again the test sample, it becomes really cruel for the especially he's going through a lot of trauma. A lot of you people are GC, the genetic counseling thing is and so on. It is really it becomes very difficult for the people to ask for the biopsy. So that's where the liquid biopsy comes into the picture. In plasma and other things, you can look for the miRNAs and other things. So we have quite a bit of panels are there. So using this technology, we can do multiple things. Just have summarized here. So in the interest of time, I'm not going to go detailed. So then I think that's with the one technology which I kind of explained, where you can do the extract the RNA and do the things. A lot of initially the in situ hybridization is one thing which people look at it, chromosome or the RNA quantification. So that is one thing. I'll just explain this this technology by just playing one video. Can you hear?
You need the audio? Just a minute.
We can't hear the music. Oh, it's okay then. It is too much. So, have you guys done the tissue sectioning and then looked at the any experiment? Have you done? So it's for example, a tissue, a cancer tissue. If you take it, first thing they do is it's called biopsy. They put it on the block called FFP block, and then they take a section of 5 micron. Then look for what are the changes have happened exactly. Okay. Okay, just leave it. Don't worry. So what you are seeing here is okay.
However, in addition to the quantitative data, social comments respect the gains and working into the tissue biology. We all understand the value of quantitative protein for gene expression of the cancer, neuron, and bone. However, in addition to the quantitative data, spatial comments respect the gains and working into the tissue biology. Spatial comments and quantitative expression data all from one slide. Demonstrates geometry. Digital spatial profiling platform. What was the context? The complex protein or gene expression profiling from just one file. A single slide for cognitive state for the creation to target areas of the tissue, which allow the geomes to be instruments to structure images with one the context. The visualization you can then select areas of interest for profiling using several approaches. By tissue morphology.
By phenotype, by individual cells, by gridding, or by contouring the area. The key to the GeoMix DSP technology lies with the photocleavable oligonucleotide tags that are attached to the antibodies via a light-sensitive linker. UV light is used to decouple the high-plex oligo tags from the antibodies, or RNA, in the selected regions of interest. Oligos are then retrieved from the surface of the tissue without disturbing the tissue, thus allowing the sample to be reused. The oligos are then deposited into a microtiter plate, where they can then be put on an analyzer for quantitative analysis. Now it is possible to get both morphological context with high profiling protein and gene expression data from just one slide sample. Experience GeoMix digital spatial profiling technology by NanoString.
So what you guys saw is now it's called spatial profiling. If you look at it, NGS, if you do it, what you are doing is RNA you are extracting from the cells and then looking for what is there. But you lose all the spatial context, exactly what is happening in the cell. So because when the development is happening, or the cancer certain progression is happening, or the immune therapy they are giving, or the therapy some drugs they are giving, in and around the tumor, what is happening, they would like to know majority of the times, or whether the therapy is feasible or not. They would like to know the tumor, whether the tumor is hot or whether the tumor is cold, whether I can give X drug or Y drug. Those kind of the information people would like to know about that. So that's where this becomes very, very important for these kind of technologies. So what, in fact, this we got it like last 6 months before to our facility. Took 3 months for us to set it up and all those things. Now we are running multiple projects based on this. So it's pretty exciting. If you look at it earlier, as soon as you dilute certain things, you are losing the information quite a bit. It's, let's say that I have 100 cells which are causing certain problematic cells in my tissue. If I don't know those 100 cells, what they are the causal agents, I'm looking at someone else there. So targets are entirely different. People are struggling with the targets, why targets are not working, why the drugs are not working. So some of these could be answered in this easily. So the PPT. So we are, in fact, as a graduate student, I always imagine, okay, can we look at inside the living cell, what exactly happens, living body, what happens. Certain scanning mechanisms are there, but at the molecular level, it's very, very difficult to look at it. So if you look at some of these technologies, the future technologies looks much more exciting, and then the really kind of a cutting edge, if you look at it. Go to the next. Okay, sorry, I can do it.
Yes.
So this is what the thing which you guys saw. So the one in the video which I explained was, let's say that I have some 20,000 or 18,000 RNA I want to probe. For each RNA, there will be a probe which is target complementary sequences created, and the DSP barcode is there. That is the key thing here is photocleavable linker, basically the UV cross linker. Similarly, for the proteins, you are looking for both here 18,000 RNA you are looking at, here 140 plus proteins you can look at it. Protein multiplexing is a real challenge because the antibodies working is a very, very challenging. So what we do is, as in the video showed, you probe them and then select the region of interest of your interest, basically using the morphological markers. In the tumor, usually we use the pan-CK, and then the immune cells, we use the CD45. So there are two markers we regularly use. There are hundreds of markers one can use based on the target biology, what they are looking at. And then finally, give the command to the system saying that please release the barcodes of the selected region. So all the other regions are untouched. Only the regions of these surround marked regions are only kind of releasing these probes. Then finally you get those probes and then count them and then look for what was happening in those. So you can do multiple ways of this selection. It could be, as earlier mentioned in the video, you can have a segmentation. You can look for certain regions. You can look for the rare cells and the contouring and also the grading. So depending on the target biology, what you are looking at cell type, you can always look. As earlier I mentioned, 226 cell types are there. Then there are multiple organs. People are doing the organ atlas using these things to find out what kind of a real genes are expressed using this technology. Because earlier, as soon as you do the bulking, a lot of information would be lost. So that's what we are. So the next is, you can almost similar to the RNA-seq analysis you can do, pathway analysis you can do, volcano plot, and then quantitative single cell deconvolution you can look at it. Then segmentation based on that you can see what is the tumor region and what was the tumor microenvironment region. Those kind of information you can get using this technology. So there are multiple panels are there. Just I'm explaining here one panel. It comprises 1,800 genes of all the tumor related genes, so that you can look for the tumor infiltration signals and then also the immune cells, whether what was happening exactly, those kind of information. Look for that. What types of tissues you can see? You can look for the FFP tissues, which I was explaining earlier. That is what the cancer a lot of the times people do. And then it's called tissue microarray block. If you can't, if the cost is, see each slide to probe, it costs almost 4.5 lakh rupees for the all the 18,000 genes to look at it. So that's where we come into the tissue microarray. If you look at 10 samples, you can put it on the one tissue microarray and then you can look for those information. So slide mountain tissue or the core needle biopsy and the fresh frozen tissue, that's what we can do. A lot of pharma companies are highly interested in these kind of technologies, and they are using this now. This is a kind of immune panel, around 100 genes are there. So since many of the biologists are interested in what happens in for the immune genes in and around the changes or the infection, what is happening. So that's where we use this panel. Depending on the target biology, you can use multiple targets, morphological markers. There is hundreds of markers are there. This is more towards the cancer connected thing, but hundreds of markers are available to you. So this is some examples of what type of publications are coming, and then so people are setting the ground rules for these things. So what we are seeing is GeoMx is a digital spatial profiler. Next one is coming as a single cell level also. That's more towards the basic research. This is more towards the screening techniques. These are some of the examples of the data which they have kind of published, how the progression of the cancer is happening. That is looked into this using this technology. This is the latest publication in the Nature Communication 22. So there are like hundreds of papers are there in that. This is another protein panels. If you look at for the immuno-oncology proteins, around 140 plus proteins, you can look at it. Each time you need to do the core biology, core protein profiling panels, and then you can always add the multiple modules depending on the interest of the biology. Similarly, in neural also we have 4 neural cell profiling panel and also the multiple modules. So that will provide you the actual numbers or actual counts of what are the number of proteins were available in and around the region of interest. So this is just a glimpse of the 2019, the DSP kind of a technology kind of launched. It was considered as one of the top innovations in 2020. So the number of published, hundreds of publications are there. So just to show the glimpse here. So we work with NanoString closely because see, there are a lot of techniques which kind of gives you the bulk information, and then which is not going to be useful for a lot of the clinical applications. So that's where we are working with them closely. So around 6,000 plus publications are there, 1,000 plus discoveries are that have been made. So we have around 125 plus clients regularly. We are working with them. Dr. Sudesh also worked with us earlier in the University of Madras, and then Dr. Munirajan. Next week, sometime we might be going and meeting Munirajan to explain this technology. So it's very interesting. So it's I think we are all excited with what the new outcome is going to be with this kind of so thank you for hearing. And just I would like to tell, like, there are 4 or 5 good students came from here, VIT. Then they're all doing very good in their thing. Aishna was there, Shriya was there earlier. Then there were some 3-4 students, and then my daughter also studied here, and then she finished that. Thanks for giving me opportunity. I'd be happy to take questions.
Now we would be open for questions. If anyone from the audience would like to ask.
Hello, sir. Sir, like we have talked about the miRNA and their importance in the diagnostics. So I wanted to ask, like, can we see the perspective, like any prospect of miRNA, like in the therapeutics also? Like, can we use it as therapeutic alternatives?
It's already being used. There are a lot of clinical trials are happening. In the US, there are 4 or 5 companies who are kind of approved also certain things. If you look at siRNA therapeutics is being used. Similarly, miRNA, one of the challenges, unless you have a perfect, or what do you call, they might have an off-target effect. So as a therapeutics, oncomirs are kind of being approved quite a bit compared to the overexpression. In the cancer, there are 2 types of mirs are there, suppressor mirs and then the oncomirs. So both could be used.
As much like I know and as much I've read, I like what I could think that if we somehow we can learn to like target, or somehow we can target the miRNA, like if we some kind of, like either we can regulate it or somehow we can like introduce it, then maybe we can use it as a like cancer stem to regulate or inhibit the growth of cancer stem cells. But so then there is one of the major problem is the delivery method, like how to deliver that particular thing, because we have like discussed so many times. So what do you think, like how can we basically deliver?
There are certain technologies like nanoparticles are being used to deliver. And then another one is certain people are very interested in the exosomes. If you guys look at it, exosomes contains certain miRNA. They act like a cargo for one end to go to another end to deliver. So people are trying to look at how do we mimic this exosomal thing, and then can we create certain exosomes synthetically and then add up this so that they will not get degraded. Again, one thing is miRNA is less degradable compared to the other mRNA kind of thing. So miRNA is already kind of 21 nucleotide. So it's very easy to go blood-brain barrier and all those things. In addition to this, people are kind of encapsulating in certain nanoparticles as well as exosomes. These are the things.
One last question, like from my side. And so, like as a student of MSc Biomedical Genetics, Like what opportunities I can see in your company, like if I'm looking for the experimental biology or not experimental, like or the bioinformatics side, what kind of opportunities can be like we can look forward in your company or the in your facility, sir?
So in our organizations, we have some 3-4 departments. One is R&D, pure R&D, where we try to develop certain products. That depends on the funding things. Another division is more on the services based on the client's project. That will be constantly, it will be there, so that the business would be continuously running. Another one could be, a lot of the times in the future, we may look for the project management is one thing which people we need always. The project management positions are less in number if you look at it compared to the overall other team. Another one would be technical sales, if you see that. So this is another thing. Then the pure sales, you need to be really hard. You have to run around and then do this stuff. And then the technical sales would be go with the salesperson whenever there is a requirement. So it depends on your knowledge level and then how you can interact with the scientists. These 4-5 divisions we always have.
Any
Thank
more
you, sir.
questions?
Hello, sir. I'm Anchal. So I wanted to ask, I am continuing in genetic counseling. So I will have to deal with patients and I'll have to help them diagnose some diseases. Okay. So whatever explanation I got it from here, I'm assuming this technique will be a little expensive. Yeah. So expensive. Yeah. So I wanted to ask, why would anyone prefer this over already existing techniques in the market?
So this is what I am telling here is it's more on the future techniques.
Yes.
All of them have not come into the market yet.
Yeah.
So in the future, that spatial profiling which I'm telling. So you have to give an option for a patient. So let's say that liquid biopsy, for example, or somebody has a cancer. Let's look at this. He has an option to go through a genomics studies or the some of the technologies that we are telling, or just say that chemotherapy. Affordability is one thing which always you have to look at it. But if you don't even tell them what are the options are, basically you are doing a wrong thing.
Yes.
Okay. But nowadays, what obviously doctors will look at, okay, whether he's able to afford. If I give him the hope of saying that, okay, you spend 1 lakh rupees for this test, and afterwards, if he doesn't get anything, he will also feel guilty. No? Because doctor has to be convinced, okay, this is absolutely, it is going to be useful for the patient. Then only the doctor will suggest. So every technologies, if you look at it, especially in the NGS, has gone a little bit ahead of this other technologies. But the multiplexing would be kind of a RNA in situ hybridization. For example, one fish, if you have to do 5,000 rupees, they take.
Yes.
Fish test usually.
Yes.
Imagine I have to do 10 fish tests for the 10 markers. It individually costs 20,000, 50,000 rupees. And just a number I'm telling. If you develop it for all the 10 markers useful, 15,000 rupees if I develop a test, is it going to be useful for the patient? That's where the the doctor has to make a call. Instead of me telling 10 fish tests, can I give him an option of one? There is a possibility that 80% you might get a result from 2 fish tests. But if I do this, the third one, there's a 100% chance. You like to go for 15,000 rupees test or 5,000. You have to give an option for that.
Okay.
Patient. Then he can take a call whether it is. If you don't give an option, then you are denying a kind of a choice for him to do this. That's what I believe. Any patient has to be appropriately mentioned what is the cost and benefit. Yeah, take a call.
Okay. Thank you, sir.
So as we talked about the convenience and the price, so like there are different companies in the market in the diagnostic fields. I had the opportunity to work with Dr. Lal Path Labs. So, sir, as I have seen, like there are panels already panels in which we have made the combination of tests and combination of the methods, and which are providing the solutions. Like as you said that we have like if we take multiple markers, and then we have to do multiple fish or the other diagnostics. So there are multiple of the panels in which there are multiple markers including and the multiple test methods are also available. Like as I remember, there is one panel from the Dr. Lal Path Labs in which we call the 50 genes panels. So we take the 50 genes and we do the PCR, NGS and all, and then we check the mutations. In the same way, we take the breast cancer panel and all those panels in which we do the IHC, and then we go for the fish also, and then we go for the NGS also. So, sir, and when we see the cost, it's not that much. Like when I talk about the 50 gene panel, the cost is around 15,000. When I talk about the breast cancer panel, the cost comes around almost 3,500 or 5,000. So when we talk about this thing, obviously it will be more than the like 25,000 or 50. Like as I can assume, like NGS cost the whole exam sequencing we go, like, then it costs 42,000 MRP. So then it will cost more than that. So then how can we justify it to the doctors?
No, that's what I'm telling you. So this is not a technology for the patient at this. Okay. This is the pharma companies are using to find out whether the drug is going to be useful or not. Why that their drug didn't work. So they work with the doctors. See, some of these tests are not tests, assays. Many of the times the doctors will do because they know the survival and non-survival information of it. Let's say that the doctor is working in a, since you are Lalpat lab, let TMH Mumbai. Let's look at this way. He will have hundreds of patient history now, and he will know only 10 patients survived or 90 patients didn't survive for some reason. He knows the drug, whatever the drugs were given, all the information. Now he goes back and look at it. Why these people? I could have saved these 90 people, or at least out of that 40 people, if I knew that what was happening. See, ignorance is sometimes the bliss. Sometimes it is not. So it's because if you know, okay, you are affordable, and then if that patient the doctor didn't tell you the options, then you feel bad, isn't it? You have to take a call on by checking with the thing. You can't judge everyone as a poor or everyone as a rich, or you have to give an option for that. That's where the people outside US and all, people spend $3,000 to look at it certain things, because some places insurance pays for them, some places insurance doesn't pay for them. How do you decide? So doctor has to decide.
So do you think in the future with more advancement and more thing, it can be used as a diagnostic?
For example, the issues which I showed, the TMA, the block, we are working with certain clinicians where they send 20 samples together on a block. So then the cost is almost equal to the normal RNA-seq work. So we always find an alternative, isn't it? But can you ask the patient to wait for 3 months for that? You have to take a call on that. So it's not, again, we are not making a decision based on this. It's in the clinical stage. People are trying to use this. Why their drug? Can they do the drug repurposing? If you have there's a test called companion test, companion diagnostic. All of you must have heard it, isn't it? There is AstraZeneca produces a thing called gefitinib, which is for the lung cancer. After a certain time, that becomes a resistant. The cells become resistant for that. If you keep on giving that drug to the patient, which costs each dose 50,000 rupees or 60,000 rupees, it's a curse indirectly, and you are spoiling the health of the patient also. So sometimes, if they're not, or after certain time, the lung cancer patient, they take the biopsy and they look for there's a mutation called T790M mutation. If that is there, they should not be giving this drug. They have to give another drug. So that you have to take, the doctors have to be advised, or the doctor will, sooner they will be advised. They will start taking a call. And sometimes the pharma companies sponsor these tests. You must be knowing it. So in such a cases, so they know that, okay, if patient can't afford, at least we will make sure that we will not give a wrong drug to them. Thanks.
Any more questions?
Good morning, sir. So like you said that exosomes are, they are able to cross the blood-brain barrier. So how promising it is to use exosome-derived microRNA for, therapeutics related to neurodegenerative diseases?
I think there are multiple diseases people are looking at. One is, a company which is being on the eye test, eye stuff, and then the cancer they are looking at quite a bit. Neuro, I'm not sure. I have not. Maybe I may have to look at it, but at least it's quite promising. But how do you? One of the challenge with the exosomes, whatever they are trying to extract and all those things is preparation of exosomal samples are becoming a cumbersome. So you have to do ultracentrifugation, or we have certain antibody-based capture kits are there. There are certain biotech companies they produce that. But having said that, in the future, if you want to have a mass scale production of the exosomes, it should mimic similar to the exosomes of the internal thing. Then it should also kind of like nanoparticles from outside can be produced that. So that might be one option. People have to look at it. But neuro, I'm not sure. I may have
Okay.
at least cancer and then the eye and other things.
Okay. So another question, sir. So when we say liquid biopsy, so we can also use a cfDNA for diagnosis, right?
Yeah.
So why would we prefer a microRNA in diagnosis?
So that's what I was telling you. So some of the thing is microRNA is kind of a 21 nucleotide. It can survive in multiple conditions, and it's an intact thing. Let's say that in the urine, we are finding exosomes are much more easy to capture in the urine and all. Especially some of the kidney diseases we are looking at, miRNAs becomes much more easy to quantify compared to the cfDNA. cfDNA only in certain cases like the cancer mutations. Those things are useful for the cfDNA.
Okay.
But not in the case of certain other diseases where they don't have any direct correlation with the DNA mutation. So these there you have to use as a surrogate markers.
So in cases of modifications, post-transcriptional or translational, then we use microRNA. Otherwise, we use cfDNA, is it?
Yeah. cfDNA, you need to know. So, see, the cancer, you know the mutations, isn't it? There are some 500 genes mutations regularly they do the panel. There's a cancer panel is there. But those pieces, if it is there in the cfDNA, then it is useful for you. The assumption is, okay, there are certain it's called tumor mutation burden. Depending on the tumor mutation burden, certain cfDNA also will be seen.
Okay.
But in the normal, let's say that there is a diabetic person who is no way connected to the mutations in the DNA. How will you look at it? So that will not be, that particular gene will not be coming. You need to have multiple mutations to look for, okay, there is at least some mutations I'm seeing. Based on that, there will be. It's a pieces of the DNA you are seeing in the cfDNA, not the certain circulating tumor cells are there, CTCs they call, but they are very less. The full DNA, if you are extracting, it is like a broken pieces. That's what it is.
Thank you, sir.
Any more questions?
Thank you, Dr. Gopi, to introduce both a wonderful technique. I think it might be very useful for the student. I know that Geomax is doing wonderful for the last few years, that every month we are getting a very big paper mapping the spatial biology of general physiology, starts from brain atlas, even the bigger projects like mapping the human brain, entire spatial gene expression of a brain. So Geomax is like, it's creating a entire new environment for that. But still, Geomax is also, the expression also based on the same encounter principle, or
there are two things. Sorry, I didn't explain there.
Uh.
Depending on the number of genes, let's say that you have a less than 800 genes, I can always look at in the encounter. If you go above the 800 genes, we are doing the whole transcriptome here.
Yeah.
We didn't show all that panels. There's a thing called whole transcriptome at last. That is 18,000 genes in the human and then 22,000 genes in the mouse.
Okay.
That has to be, barcodes will be kind of amplified and then prepared library. Then that is sequenced in the NGS machine. The other ones, lesser the panel, you can always do in the encounter.
So after relieving from the linkers, either you can go for encounter or you can go for NGS based on a number of things. Okay. This is an independent mission. It just does the spatial thing only.
Correct.
Then you relieve it and you use whatever your technique you want. You can
It's
use.
an independent. We have it has a server which comprises the DAS. It's called a DSP analysis suite, basically. As soon as you plug the other data, it automatically converts into DCC files, and then the NGS files are converted into DCC file. Finally, you look at which ROI from the data was coming. All those things analyzed based on.
When we talk about encounter itself, we already discussed that how much efficient this CCD sensor should be to pick up each individual nano signals, because we are not amplifying, right?
Yeah.
We're just capturing it single molecules, and the imaging should be so good. How long it's taking for taking the image? Because the problem we face in the imaging is
usually the whole run on the cartridge and then the processing is 6 hours. So 6 hours. So the 3 hours typically the processing happens, the washing and other thing, and then the 3 hours for the processing those 12 lanes.
A quick question is coming from our online participants,
Sure.
sir. So RNA biology happens to be an up and coming area of research. How is Theracuse contributing towards facilitating research?
So, good question. Yeah, as I said earlier, if you look at the FFP samples and all those things, it was very difficult to do in the normal techniques. So we are the first company to get this technology, and then 2017 we started working in this thing. And then we have around like 10-12 publications who have worked with clients. Then also there are a lot of pharma companies we work with closely. So we are trying to hasten some of precision medicine should be more like can we develop certain companion diagnostics assays? So that's where we are working on that. As I earlier mentioned, liquid biopsy is one of the things. We are looking at the VC funding rising and all those things. So based on that, the product should be developed based on.
Thank you, sir.
Presently, we are doing more services, 80% of the service, 20% of the product development. In the future, it could be 50% service. As the revenue increases, the number of people increase, 50% could be product development team.
Thank you.
On behalf of SBST VIT Willow, we thank you, sir, for your insightful and fascinating lecture on RNA dynamics. Thank you, sir.
Thanks, sir. Thanks a lot.
I would like to invite Nikita for an important announcement.
Before we break for tea, I would like to inform all the students who haven't registered for treasure hunt can come and give the name at the entrance. There'll be 2 volunteers sitting over here. So whoever haven't given their names, please do register. And now we break for tea and it's served outside. Make sure that no food, no eatables are allowed inside the gallery. And we resume back at 11:30 again in this auditorium. Thank you.
you all wholeheartedly to attend this workshop organized on the occasion of 9th National DNA Day. We take the opportunity to welcome and introduce Dr. M. Rajni Raja sir.
Dr. Rajni Raja is a faculty from the Department of Biotechnology at the School of Biosciences and Technology, VIT Vellore. He has done his PhD from VIT in biotechnology and did his Masters of Technology in Bioinformatics from Sastra University. His major area of research includes drug design, molecular modeling, and bioinformatics. His publications on drug design and in silico analysis are quite insightful and has left a major impact. Now, we would like to invite Sir on the dais and enlighten us with his knowledge and wisdom.
So, very good morning. Very good morning to one and all. So, as you all familiar with me, since I am from the same university. So today's my topic is basic in molecular modeling. So molecular modeling, till now what you had from the previous speakers, all related to an experimental model, experimental approaches, how the DNA, RNAs are, studied, okay, experimentally how it is studied. So now I'm going to talk about the molecular modeling with the help of computer approach, So computationally, how do we handle the molecule? Okay, so that is what my lecture. So molecular modeling, the molecule, I would say that the molecule can be categorized with respect to the size. It is micro as well as the macromolecule, right? Can you guess what are the micromolecules? Micromolecules from your biology. Micro means what? It's a small molecule, right? The size is very tiny, or maybe we can start from the atom, right? So when you come to the macromolecule, it may be a RNA or a DNA molecule. Okay, so instead, one step forward, we have a protein molecule. When you talk about protein molecule, we can express, or we can identify the mechanism about the protein molecule. Okay, so computationally, how do we handle the protein molecule with some techniques? Okay, that is what today's lecture. So here are the topics what I have for the discussion. Start with the introduction and molecular mechanics model and macromolecular structure prediction and validation. If time permits, we discuss little about the molecular dynamics simulation. Okay, so before I start this, I would briefly discuss about how the biological and chemical process are happening. You know that you people are studying with the various experimental approach, right? How do we take it into a computational approach, and then we can apply this into society, right? So here are some examples I displayed here. In the left hand side, you could see the biological and chemical processes from the cell, right? Inside the cell, we have several proteins or enzymes are always working. And also, you can have a kind of organism which are having a variety of protein molecule, which may be a target for a particular disease, right? So experimentally, though you study, I have some techniques to identify the mechanism of
action of a particular biomolecules computationally so that I can use it for the applications like therapeutic process or a biotechnology applications like, bio nano device development and new drug. And we have so many applications with the help of the computational approach. The understanding here is the difference between the experimental and theoretical or a computational approach, right? So you people are studying experimentally something, but how do we take it into computational approach? Okay, so every experimental approach have their own computational approach, but we need to understand the basic of how the experimental approaches are happening. Then how do we understand the basic physical model of the particular experiment, and with the help of mathematical equation and the physical model, we are computationally analyzing the molecule with the help of tools and software. Okay, so when we invent the tool and software, we have the basic understanding of how the experimental data are working and what are the basic components required for the experimental process. How do we link this data into the computational approach? And this approach, how do we take it into a application process, like developing a drugs, etc. Even we had a vaccine recently for the COVID-19, right? Within a shorter duration of time. So with the help of this computational approach, we could be able to achieve this. So this is what the small comparison between the experimental and the computational approaches. But then there are some limitations to handle the biomolecules with the help of computational approaches. We'll discuss some examples of this biomolecules computationally, how it could be analyzed. So generally, the computational analysis takes like, for solving any kind of biological problem, the particular problem, you do have the physical model with the help of this mathematical formula. We do have the software or a tool for solving this problem with the help of the computer, right? So it is all interlinked. It is not a single field to achieve this task. So we need to thoroughly understand about the basics of the whatever you are doing the experimental process. So here are the information about that. So coming to the molecular, micromolecular mechanical model, that micromolecule, if I take the micromolecule, it can be started from the atom, right? From the scratch. So we don't know anything about what it is there. So I'm going to start from the atom. So as I already told that the molecules, the biomolecules has been separated by two categories, that is micro as well as the macromolecule. So micromolecules are having some specification for analysis using computational approaches. So here the micromolecules, further can be divided into two. Okay, so of course, this is a small molecule, but then we have a different level of understanding. So start from the quantum mechanical model. Further, we have the molecular mechanical model. You people are studying from the quantum mechanics of a particular molecule. It starts from the electron, proton, combination of both electron and proton. Sometimes it is nucleus and proton or nucleus and electron. So many theorems are there to understand. See, here are, in a nutshell, I can show you the entire quantum mechanical model, right? Start from the Schrodinger wave equation. This is going to give you the energy of a particular molecule, right? So the wave function, that is how we have started with H psi equal to E psi, right? And the Born-Oppenheimer approximation is going to be a combination of both electron and nucleus. Likewise, you have several approximation method to reach into finding out the best approximation of energy, right? So everything deals with finding the energy of a molecule. So quantum mechanically, we have several stages to understand the the mechanism of the particular atoms in terms of electron or proton, and we reaches into the energy. That is what the quantum mechanical approaches, but we are not going to discuss more about this. But when we come to the molecular mechanics or a classical mechanics, okay, so we have some the reduction from the quantum mechanical approaches. We are neglecting the electron terms, and we are using the valency and bonding concept. With the help of this, we do understand about the molecular mechanical model. So if you take a molecule, we have several properties to understand. So here, if you take a molecule, say for example, water molecule, we have displayed in the right hand side. Maybe it is having 3 atoms. You go with more than 3 atoms, we could have more number of the properties or parameters to understand. So initially it starts with the stretching of energy. The stretches which is followed by the Hooke's law, and we have the bending, bond bending between 3 atoms, and the torsion, the the movement between the 4 atoms, and electrostatic interactions between the atoms and van der Waals. So these are the basic parameters which we always go for finding the energy in terms of the molecule, right? So if we move into the molecular level, so we need to understand all this energy term, right? So bond, so this with this 5, if you take an example of some compounds, the summation of energy of a molecular system is very long equations. If you find out the energy of a particular molecule, which is displayed in the block color box, right, having mixed with the carbon and hydrogen, oxygen, with a different type of bonding, right? So if I wanted to analyze the total energy, the potential energy of a molecule, so I need to go for different parameters like stretching. You could see the stretching between the atoms. The list goes, right? And the bending, torsion, van der Waals, etc. So these parameters are very useful for finding out the total energy. So this is how in the molecular level analysis with the help of computational approaches. There are several force fields are there for finding out the energy. It is available ready-made in the market. You can choose any kind of force field that can be used for predicting the energy of a particular molecule. So here are the list of the force fields are available. Some of them are very familiar. You can take an amber, charm. So these are all the familiar force field methods. So it is just going to predict the energy with the help of these parameters, whatever I told in the previous slides. So here in deep, when you explore the amber, you have the several terms which I already explored in the previous slides. So the equations, the mathematical equation for finding out the energy. So this formula has given here with respect to the stretching, bending, and torsion, and some of the cross terms are there, and van der Waals and electrostatic interaction. So all put together, you have a total potential energy. This is how the calculation of energy goes for a smaller molecule. If you take a small molecule, even if you are working for ligand preparations, or you are designing some drugs, So you need to go for understanding the energy, to the total energy, with the help of this kind of, the force field methods. So with respect to the force field, the restricted number of atoms in each category we have given here. So the number of atom types increases, which is very easy for analyzing the molecules, the bigger molecules easily. So for example, if I have maximum number of 213 atom types, so it may be analyzed a bigger size of molecule. Okay, so if it is 40 atom types, it will be analyzed only a small sizes of molecules. So based on the type of force fields which we are selecting, the analysis goes. So some of the the force fields are embedded in the software or a tool which are commercially available. So this may be useful for analyzing or finding out the energy. Okay, so this is how deals everything deals with finding out the stable energy conformations in order to minimize the energy. So here the molecules are always moving. We don't know about the nature of a molecule. The each and every fraction of seconds, the energy levels are varying. That has been represented in the middle. This is the potential energy surface of a molecule that is not stable. So it may be a the wavy-like structure because of the movement of the molecules, right? Here we have to see about the potential energy term. We are not moving the molecule, but then it is moving themselves to adjust the environment surrounded by the molecules, right? So here, this is how the small molecular analysis goes, right? So till now, what I talked related to the small molecular analysis, right? So if you start with the atom level and move on to the small molecules, so all this comes under the molecular mechanics. So now I move into the macromolecular approaches. So macromolecules also keep moving in every fraction of second, right? So before going to see how the behavior of the molecule, we could have the prediction of the molecule computationally, how it is happening, why we need to go for macromolecular prediction, and after the prediction, what would be the validation of the molecule. So once you do all this thing, then you can study about the dynamics or the movement of the molecules and their respective, the stability, all this thing you could be able to study. So I'm going to concentrate only on how do we predict the macromolecules. So you have so many applications in the macromolecules, but computationally, this is the hot area where the people are working for modeling the protein and finding out the mechanism of the interaction between the other molecules. So we need to have at least the structure so that we may be able to understand the behavior as well as the mechanism of the macromolecules. So when I say macromolecule, it is completely devoted to a DNA or a protein, right? Mostly we are talking about protein because DNA it is quite difficult to handle with the help of computer, but then we used to highlight or show the structure with the help of computer, but it is quite difficult to find out the interaction between the DNA and the protein. But whereas protein, we have a separate database for that. There are some methods for experimentally crystallizing the protein structure and deposit it into the PDB database. So we have PDB database for that. So from that, we may be able to get some useful information and we can predict the molecule for the unknown protein structure. So this is the hot area. Why we have the modeling always? You know, what would be the limitation of the experimental method? So this is very important. You know, the available protein structure in the database, PDB database, with some number, right? But the annotated protein sequences present in the protein sequence database. You know, I don't know how many of you are aware of these databases and all, right? The sequence database, there are separate database for maintaining the sequence data and structural data. You know, we have so many data, right? So this structural data is very minimal and the sequence data is the higher. We need to compete this the gap between these numbers, right? So we have the limited number of structures because of the complexity of the experimental methods. You know, what is the method for use for protein crystallography? How do we identify the protein structure? What are the methods we are using for identifying protein structure experimentally? NMR, X-ray, very good. NMR and X-ray. You know, the process of finding out the structure of a single molecule, there are several stages, right? So that's why it is very difficult to crystallize all the sequences which are identified and deposited into the protein sequence databases. So how do we overcome this gap? How do we equalize this gap? So we have some computational approaches to identify the molecule which are unknown with the structures. Okay, so the protein sequence whose structures does not available in the PDB database. For that, we are going to model. Okay, so of course, we are going to get the information from the PDB database. So this is what the objective of this, you know, the 3D structure prediction. So we are unraveling the 3D structure of protein sequence whose structure not been detected. Okay, so so computationally how it is possible. Okay, that's what we are going to see. So here are some sequence which whose structure does not there. Okay, so we have sequence with something, but we don't have the structure for the particular sequences, right? So how do we make a particular 3D structure for the given unknown sequences? Okay, so with the help of some algorithm, we may able to solve this task, right? So what would be the algorithm? That is what we are going to see in the next slide. So here the approaches to predict protein structure, we may always start with the target sequence, right? So you have the target sequence from the the database for which you are going to model the structure. Okay, so if I model the structure, so we need to have some informations about the folding data. So where do I get the folding informations? So from the existing database. Okay, so the structure is always available in the PDB databases. So for my target sequence, I can compare my sequence with the structure database and get the folding of the protein. Okay, so for after getting the folding, we need to segregate the methods based on the type of similarities between your target sequence and the folded templates. Okay, there are some sequence similarity between these two. So this is going to decide what method you are going to choose, right? So here the homology or comparative modeling and the threading, these are all the knowledge-based method. The knowledge-based, nothing but we are taking the information from the existing knowledge. Existing knowledge in the sense it is available in the database, right? So the other method we have the sequence whose structure is not there in the database. So sometimes, it is also possible for the sequence, you may not always have the structure in the database, right? Sometimes we may not have the structure. So for that case, we go for ab initio modeling. So it is going to be start from the scratch, right? So finally, using these three approaches, you will be reaches into the model, and that is used for validation and then further process. So this is how the complete, the predictions of the macromolecules goes. So here are the comparison between these three models based on the similarity I told you in the previous slide. How do we select or specific method for the sequences? Here under the homology category, it requires at least greater than 30% of the identical sequences. The between the protein and target sequence, we need to have some percentage of identity. So if it is more than 30%, then we can go with the homology modeling approach. Okay. So if it is below 30%, or maybe between 10 to 30% range, then we can go for fold recognition method. If it is less than 10%, then we can go for ab initio approaches. So we have different approaches based on the percentage of identity and similarity. You can select the particular methods and you can go for it, right? So what is a homology modeling here? That is what this slide is going to give you. So it is a prediction of 3D structure of a given protein sequence based on an alignment to one or more known protein structures. Okay, that we have already been discussing. So if the similarity between the target sequence and the template is detected, then similarity can be assumed. So it can be used to understand function, activity, and relationship once you model. Okay, so now we have a different understanding of how to do a homology model. So here we have the target sequence whose structure is not known. Okay, but we have some structures in the databases and the respective sequences also there. So when we compare these two sequences, so what would be the limitation if it is a homology model? It should have about 30% identity or similarity. So if 30% identity between this target and the template, then I can take this structure. This is the known structure for the respective model, and I can take this model and then model it for my target sequence. Okay, so here the homology model takes greater than 30% of the identity, right? So here the comparison between these two, target and template, this is available in the database, but this is we may not know about the structure. Okay, so if the 30% identity is there, then you can model this particular template into your target. Okay, so there are some algorithms are there for modeling the protein structures, right? Yes or no? So here are several stages of homology model. So each and every steps, they are following some algorithm to solve the task. Okay, now there are several online softwares are available So your job is to upload your target sequence. That's all. So it is going to predict all these steps. What it is, you can start from the beginning, that is template identification. Okay, so template identification, it is going to identify the template from the PDB, and followed by we have alignment corrections between target and template, and backbone generation, and loop model, sidechain model, model optimization, and model validation. So in the right hand side, we have the pictorial representation of how do we take a molecule. There is a unknown protein sequence whose structure is not known, and we are comparing the sequences of the known sequence, known structure, and we are finalizing the template. Then we combine these two to generate a model, and then validating your model. So this is how the homology modeling steps goes, right? So, so here each and every step has been explained clearly here. I don't know how many of you are aware of this homology model. Does anybody have done this before? Yes? Okay. So let us quickly go through all the steps. So the first step is that the identification of suitable template, right, for our target sequence. So this is achieved by the comparison tool, that is BLAST, that is basic local alignment search tool. See, if I find out the template for the given target sequence, the BLAST is going to identify the template from the database by comparing the target sequence with the existing PDB database, right? So when we see the result of the BLAST, we have the list of hits which are highly similar with your target sequence, right? So we cannot blindly select our template, right, based on the order which they have listed here. I cannot take the first one because of the percentage of identity, right? So I have to choose my best template based on these criteria. So I have to look out all these criteria. Once the particular hits passes all the criteria, then I can choose the particular template for my homology model. So what are the criteria? The first one is the percentage of identity and similarity. That is obvious, right? So how much I required for modeling with the help of homology model? More than 30%, right? So 30% or more than that, we have enough for the homology model. But here you can have so many, the hits with the different sizes or different percentage of identity. Okay. If I clearly mention, the first one is the 67%. So how many are eligible here? The first 6 are eligible based on the percentage of identity, because till 30% I can choose. So the second criteria is the query coverage. The query length is represented here. It is nearly 180. The template also should cover the entire region, so that we may not lose any part of structures for our target sequence. So the query coverage also very important. Here the first one covers only the 1/3. Okay, here it is 1/4. So these 3 are maximum coverage, having maximum coverage. So I cannot choose this because of the identity. I cannot choose the first one because it does not cover the entire region. So that is what we need to follow, the query coverage. And the third one is the family of protein or homologous features of the protein. So my target sequence in the name of nicotinamidase or pyrazinamidase, whether the hits also have the same kind of family or a homologous. So homologous means the same functional gene present in different organism or a same organism, right? So that is also very important. The other two are very less consideration, but first three is very important. Okay, so this is how you have to choose the best template. So once the why I clearly explained the selection of template is the initial stage is very important. If you choose the wrong template, then you may reaches into the false result. So the selection of template is very important. That's what I given the criteria for the selection. So in addition to that, after selecting the template, you can look for other properties like whether the molecules or templates bound with the ligand or what would be the resolution. Okay, so all these features you have to check. Okay, so all the other things are preliminary. Just I quit this because everything has been done with the algorithm. Computationally, it is going to be performed. But if time permits, you can look for the different stages of how the homology models are going, right? So I think the bioinformatics students can understand about the homology model. You might study in the future courses also, right? So I'll just skip all this thing. I'll just read the steps. Okay, so this is the backbone generation between the target and template. And this is the loop model. Wherever the insertion and deletion between the target and template, we need to follow the looping procedure. So the pictorial representation is displayed here. Once you generate the backbone, then you are going to attach the side chain of all the amino acid. So side chain modeling is happening separately. So everything is, they have their own algorithm. Okay, you are not going to do anything. So everything computer is going to do. Okay, but you need to understand at least what it is happening behind the screen. So here the side chain modeling based on the molecular optimization methods, it is going to adjust the molecule so as to bring the more stable conformations. Okay, so they are following some principle for optimizing the molecule and arranging in the PDB structures. So the previous stage we are optimizing in the sidechain model, but then as a whole we have the optimization technique to minimize the energy, right? So it is going to arrange the molecule in order to increase the stability of the molecule, right? So that is what it is happening here. That is model optimization and followed by the validation. So once you achieve the model, your job is to validate the model. You know, some servers are doing this process, but most of the servers are not doing this process. That is a validation process. You have to manually do and check the quality of your model, right? So there are some servers are there. Okay. So this ProCheck, WhatIf, and ProSA and Verify 3D, all this are available in one site, that is SAFE, that is Structure Realization Verification Server, where you can upload your modeled structure and you can check the quality. So this is one of the major quality which we are always check after the model, that is Ramachandran plot, right? So the Ramachandran plot, it talks about the phi and psi angle between the peptide backbone. You know, the peptide backbone, we have several atoms, right? I think you could see, you would be studied in molecular biology, right? So when a protein fold, the stability can be increased by keeping or arranging the molecules in such a way that the stability could be increased Generally the globular protein, the hydrophilic molecules are placed in the surface and hydrophobic in the core region, right? But we are not going to talk about hydrophilic or hydrophobic, but with respect to the secondary structural element, that is alpha helix and beta sheet, we have categorized here. This part is going to take about the beta sheet. The phi and psi angle of the respective amino acid should fall in this particular region. Okay, especially for the beta sheet. Same way we have left-handed and right-handed alpha helices, right? So this quality is very important. The model should, whatever you are modeling, the modeled protein should be arranged in such a way that it should fall in this particular region, right? So the red color one is the highly or a core region. Okay, the yellow color is the allowed region. The white one is the disallowed region, right? If anything comes down into the white region, then your job is to bring the particular amino acid into core region by doing energy minimization, or sometime you have to do some loop modeling also. So you have to identify the position of the particular amino acid and then take it into a core region by loop modeling. So the quality should be checked with the Ramachandran plot. Okay, that maybe during the demo I will show you. So this is how the summary of the homology model. Okay, first we need to identify the suitable template, and you have to create the alignment between target and template, and you have to perform the homology model. Okay, and followed by you have to analyze your model structure and validate the structure. And if it is anything goes below the quality, then you have to refine the molecule. So this is how the strategies for the homology model. Okay, so it's important to take time when constructing a model. Crystallography is difficult and time consuming. A model should not be rushed and should be fully checked. So it's not like uploading the sequence and getting the model structure, your job is over. So you have to check your modeled structure and see the arrangement of atom and validate your results so that it reaches into the next level of research, right? The model, the one which can be taken into docking, right? So before going to that, you should check all the quality. So the next step is here is the mechanics comes, right? The simulation, molecular dynamic simulation. There are several stages are there in the molecular dynamic simulation. So for the sake of time factor, I'm just skipping this. I just end the session. So micro and macromolecules are not static in structure as how the X-ray crystallography suggests, but it is continuously moving. So we need to understand the behavior of the molecule, and it should not exceed the boundary of the the space. So here are some of the example whose dynamics given here. So this is how the analysis goes. Thank you. So I would like to thank the organizer for giving me this opportunity, especially Dr. Radha Saraswati Madam. So I'm happy to get some question from you, if anything you have. Shall we move to the demo? No doubt. Okay, thank you.
On behalf of SBST and the participants present here, we would like to thank you, sir, for such an informative session. We are really delighted to have a session full of knowledge. Thank you. Thank you so much, sir.
Now, I request all of you to move to SMVG 15 for the hands-on session. So, yes. Yes, attendance will be taken at SMVG 15. So please move. And also, everyone needs to be back by 1:45. We have the inaugural session at 2. So, and attendance will be taken at SMVG 15. So kindly move.
हेलो, मेरी आवाज आ रही है? वीडियो भी स्टार्ट करनी मेरे को मेरी। हेलो, आर यू एबल टू हियर? एक बार बोल के देख, लाइक हेलो, आवाज आ रही है? Okay, it's sharing now. You are able to see the Firefox screen. Okay. Yes, sir. It's present.
So, very good afternoon to all of you. So now we are going to show the demonstration of how to do the homology model through online mode. Okay. So first we require the target sequence, right, for doing homology model. Since that is the initial data,
Are everybody able to see the screen? Is it visible till the last bench? Yes. Are you able to see the screen, right? If any problem, you can tell. Okay. Yes. Thank you, sir.
So here the first job is to download the target sequence from the UniProt database where we have protein sequences. So the database is highly devoted to the protein sequences. So once you identify the target, then you go there and download the sequence, right? This is how the research goes. But for the sake of requirement of the target sequence, we have given the UniProt ID. What you do, you go to the UniProt, open the browser and type UniProt. Click that UniProt. Here you have the find your protein, right? Where you type the accession number. So that is the accession number given in the alphanumerics with the 6 characters, right? You please type here. So this is how the sequence page of the UniProt, but here you are not looking the sequences directly. In addition to that, there are other informations like function and so on. You can see in the left hand side, right? But I need to download only the sequence, right? In the form of FASTA. FASTA format sequence, which is nothing but it is started with the greater than symbol and one line description is there. Next, the sequences is starting, right? So here is the download option, right? And click that FASTA canonical. So you will get the sequence. Anybody having doubt, please stop me. Okay. Have you reached the sequence, all of you? Have you reached the sequence, all of you? Okay. So this is your target sequence. Okay. So you could see the accession number, whatever you have given here, respective sequences is opening here, right? So in the name of nicotinamidase, pyrazinamidase, in the organism Mycobacterium smegmatis, right? So the one line description is there, followed by you have amino acid characters. That is the sequence arrangement, right? So we need to take this sequence this is for our target. Okay. For this target sequence, we are going to generate the homology model, right? As I already told you that we have two different approaches. One you can do offline mode through offline mode, and other one you can do online mode also. There are a lot of servers are there, online servers. You can directly upload the sequence and get the model, right? So here time permits, I will make a small demonstration of the offline mode using modeler, but now I can show you how to do in online mode. Okay, so all of you go to or you can freshly type the Swiss model. Swiss model. Switzerland. Yes.
Should I hide this?
Yeah, that's what. Thank you. So here you could find the first hyperlink, that is this model. This is the online server, right? So you can just click that. Please do follow me and start modeling, right? So everything is by clicking mouse. Okay, right? So this is the Swiss model server where you have to paste your target sequence for which you are going to get the model, right? Yes or no? So here are the box, the text box where you have to paste the sequence. Can you try? So whatever the sequence you have downloaded, you please copy it and paste it over here. Yes. So here you have the option to give the title of your work, right? So if you want, you can give your title of the project. For example, homology model, right? Okay. So followed by you have two hyperlinks are there. The first one is search for template and the next one is build model, right? So unless until you are specific to a particular template, you cannot go the first link because it can choose their own template and model, right? If you have any knowledge about the template, then you can find the template and you can find it whether the right template what you are thinking is coming or not. So that is how the first link, but it is not mandatory always, right? If you want, you can go and see how the list of templates for the given target sequences. So okay, so template is nothing but what? It is the known 3D structures extracted from the PDB database, right? The next one is the build model. So you can directly build a model that is going to take by default whatever the templates are available for the given target sequence and going to generate the model for you. Okay, so you just click that build model. Take some time to generate the model. Is it loaded? 40 model? See, once again I repeat, the sequence you have to get it from the UniProt database, right? Type the database name. For example, I'm repeating it. You can type it in a Google UniProt, right? And you will reach the first link. This is UniProt knowledge base. So instead of sequence, you have the other informations also related to the gene. So that is what they have given, knowledge base. So here you can type the specific gene sequence ID, which I given in the board. Okay, you type the sequence ID. This is the UniProt sequence ID. You type it here. So it is taking you into a sequence page of the UniProt, right? So here you have so many informations along with the sequence. Okay. You can scroll down and you can see the complete information related to name of the, the function of the sequence, the structural informations, family, domain, etc. So here is the sequence, right? You can download the sequence in the FASTA. Okay, so you just copy this. This is your target sequence. This is how the selection of sequences of your target interest, right? So, in the literature, you could have the details of what is the specific target. Okay, so you can get the name and you can type it in the UniProt and get the sequence, right? If not the sequence ID, at least the name of the target sequence by which you can search and get the sequence. Yes? Okay. So, so once you copy the sequence and paste it here in the Swiss model and run model. So now we got the output page. Okay. So this output is based on the quality of the results. Okay. So the list goes for some numbers. Okay. So the first hit will take the highly appropriate template. Okay. So to identify the highly appropriate template, they have given some scoring factors, right? GMQE. Okay, so they have given some of the scoring factors and the respective model also displayed here. So now our job is to download the model from this results page, right? So I think this one is having highly suitable model because of the highest score, right? So here you can click this model and download in PDB format. Okay, so this downloaded here somewhere in your system. Understood? So before going to download, you can see the template also, template informations of the particular target. Okay, so the template whose PDB ID has given here, that is 3PL1 and with the A chain, right? So it is also from the same family. The maximum coverage is there. Sequence identity is 66%. So all the information is given in the results page. So it is satisfying the first three criteria. So this is the suitable model for your target sequence. So once you download, you can view the structure with the help of visualization tool, right? So this is the model you have downloaded from the Swiss model page. Okay. I don't know whether in your system this viewing softwares are there or not. This is the Swiss PDB viewer. I think you could also try from your system. Swiss model is Kitna? Swiss model continue. Swiss prod viewer written formula. And the terminal side. Okay. It is there. You can just listen here, listen in the screen. Okay. So till now, you know how to do the modeling, right? Through online. You are downloading the target sequence and using the Swiss model, you can paste the sequence and submit the model and wait for some time, and you can download the suitable modeled protein. Now we are going to view the model with the visualization software. So this is one of the software which is going to view the protein structure, that is modeled structure, right? So here to control this structure, we have the control panel. Please listen. If you have in your system, you can try. Otherwise, you can listen, right? So here, I don't want to explain deep about this tool, but most important thing is control panel. You can get it from window control panel, right? So this is where you are going to control the entire structure. So I wanted to see the structure in ribbon mode. Okay. I'm just hiding all other things. That is the backbone and side chain information. I could see only the secondary structural information of the protein. Okay. So just we have given a target sequence and we got the 3D model, but we don't know about whether this modeled protein is having folded properly or not, right? So for which we need to go for Ramachandran plot. Here also this software is embedded with the Ramachandran plot, but your job is to select the entire atoms for this particular PDB 3D structure and go to window. Here you have the option Ramachandran plot. So this is what we have discussed during the theory session, where I have highlighted in the red color, yellow color and white region, right? But here the representation is different, right? So most of the amino acids are falling in this particular region, except if you can keep the cursor over there, and then you can see what is the type of amino acid which is coming outside the core region, right? So this is somehow, you know, affecting the quality of your structure. Understood? So it is always possible, but most of the time, you know, you may not expect this kind of amino acids. Okay, so outside the core region, only few amino acids are exempted, that is glycine, because it does not have any side chain. So there is no, it is not having a sterical hindrance. So the glycine amino acids are exempted in the outside region. But other than that, you have to see all the amino acid. Okay, this is glutamine, right? This is leucine. So likewise, you have see all the amino acids which are coming outside, right? So obviously this will affect your quality of your model. So what you do the energy minimization of this particular model, and then you try to bring those amino acids which are located in the outside region and take it into inside the core region. So if you do 2-3 times refinement, sometime it is possible to move into the core region, right? So we may not expect always moving into the inside core region. If it is not moving, then you have to do loop modeling. That is the next homology model that we are not going to discuss here. Loop model in the sense the location of this particular amino acid, where it is there in the secondary structure, and you adjust the structure preceded and succeeded by 2-3 amino acid and make a loop, so that those regions can be bring it into this region, or it may be made compact. Okay, so that may not be effect for your protein. So this is how the analysis goes, right? So I will try to show you how to refine the model. They're asking. After the model. After the model. So I'm repeating the Ramachandran plot here. So once you model the structure, you are loading into a viewing software, that is this model or this PDB viewer. This viewer is also have the option of checking the quality. Okay, but we have the other option, that is saves server, where you can put your model structure and you can check the quality. But here itself you can see the only about the Ramachandran plot informations. Okay, so before going to select this Ramachandran plot, you have to select the atoms completely in the control panel. If it is not selected, then it will not reflect into the Ramachandran plot. So before going to click, select the Ramachandran plot, you have to select the amino acids which is there in the control panel. Okay, now you can see the plot, right, where you have the arrangement of atoms. So I have highlighted in the theory session, where the left hand side corner, what is that? Beta sheets, right? Right handed and left handed alpha helices are there in the diagonal boxes. Okay, so this is how the phi and psi angle for our modeled structure. So it is mostly the properly folded protein. Understood? So I'll just quickly show you the other server. You can type SAVS structure analysis verification server. This is another server for checking the quality. Instead of SPDBV viewer, we have the other server, that is SAVS. You click so this server is controlled by UCLA, University of California, where what you do, you please select your modeled protein. Okay. Just run the program. Okay. This is what we have discussed during the theory class. You have a different level of checking the quality. Okay. Here also we have the Ramachandran plot. Start with the error, verify 3D, and what check and pro check. You click that pro check to get the Ramachandran plot. Take some time. If the server is very slow, sometimes it will be like this only. Sometimes it may be very quick. Yeah. Shall we stop? Okay. So this site is going to display the Ramachandran plot. Okay, what we have already saw in the Swiss model. Okay, Swiss PDB viewer. Sorry. So the first one is the error, which will show you the overall quality of your model structure. So 94.67, above 90 is very good model. Okay. So only thing we need to check the Ramachandran plot. It's taking time. So if you have any questions, you can please ask now itself. Till the servers is loading. If you have any questions, please ask. Coverage of the template. That is, you can see here during the yeah. So the coverage region is given here. You can keep the cursor in the coverage. It is showing the length, 1 to 179. But our target sequence is nearly 180. So it is covering maximum region. Okay. Can you see that? So our target length is, I don't know the numbers given here or not, but if you see the sequence length, it is nearly 180. See 187. So 179 is the coverage. So only the 8 amino acids are missing. So that won't affect the quality. But 100% coverage should be there. That is the best model. But sometimes we may not we may not have the data in the databases. So whatever the data it is there in the database, that only we receive. Okay, we cannot expect the 100% coverage always. Okay. Any other question? No, identity and similarity is not going to come here. So coverage is just a length. But for the entire length, how many amino acids are having identical with that template? So if it should be more than 30%. But according to this, we have 66% sequence identity. So that means it is more than sufficient. Okay. Any other?
In the deep field software, how do we change
That's what you have some practice for handling this tool. This is the control panel where you have different option, showing only the wireframe and side chain. I'm hiding the secondary structure. This is the kind of representation. We do not know about anything, but if you wanted to label the amino acid, you can label individual amino acid and you can see the location. And you can see only, you can select only the secondary structure or for a particular sheet. Okay, that is also possible. I can show only that particular secondary structure. So it is something like, handling the tool. So I think we'll come to end of the session. Due to the time factor, we need to move on to the next session. That is the inauguration at 2 o'clock. So before that, we need to take lunch. So if you have any queries, you can ask me individually, then I will help you. Thank you.
Ma'am had one question.
Oh. Others can go for your lunch. Thank you.
For the lunch, everybody for the lunch who have registered to get the lunch can take the coupon from her. And outside there will be volunteers who will come to the food for the lunch. Okay. Main canteen, lunch will be there. Take the coupons who have registered. Okay. If you have, and for the inaugural, everyone have to be present at the VOC gallery by 2. Attendance will be taken there. Attendance will be taken there again for the inauguration. So everyone should be in the VOC gallery there by 2. If you have any queries, you can ask the person.
பாருங்க டவுன்லோட் பண்ணீங்கன்னா இந்த மாதிரி எஸ்பிடி வியூவிங் வரும். இது பாருங்க. இது உள்ள போனீங்கன்னாக்கா இங்க இருக்கு. You click this, you can locally you can download and it's free.
we did start little late. It was in July. We started talking about it with the students, and then we took it up to the HOD and then our dean. There's never no from anyone. That is what I want to tell here. So students of biomedical genetics are so fortunate, right? And like she said, the legacy is going to go on. I would really love to see the 100th legacy also. So it is nice, to see when we remember Gregor Mendel this year, his 200th birth anniversary, right? I'm very happy about it because not even a day goes without thinking him. So when I say, oh, the wood characterizing the genes, everything, the clothes we wear, everything has started off from then. And now finally we talk about genetic counseling. That's also from there only, right? So what I would like to say is all biotechnologists or microbiologists, anybody in life science are connected to genetics. That is what. And then I have to thank our students, biomedical genetics. They just started off. Everything doesn't go with one student or one teacher or one faculty or one individual. It has to be a team. And I'm sure you all know, much is talked about collaboration. The same thing happened over here also. These students started working from July, August, then September came, and today's 17th September, they started. I told, oh, today the D-Day, and they just done it very, very well. Applause to all these students. Can you all? Yeah. Each of them did their best. They were just running around. You can see now also many have not come, and some of them are in the canteen, trying to push all the participants over here. This time it's a hybrid event, like all of you know. They have struggled a lot for both to be seen, and now I see Dwayne moving up and down here, So we have tried, and one more thing, with your permission, sir. Yesterday when we were talking to the CTS and all, they said, because a lot of programs are going on in VIT now, we were not able to get the camera or nor the video. Then we have to pay for it. So, but always our DNA Day goes with minimum budget. As much as possible, we keep it at low so that we don't want it to be no money should be taken from the students. That is what VIT says, and we never do that. So there are sponsors for it. So we have ILE and Ribozyme who have sponsored for this also, as well as participants all over. I think there are some 39 education institutions and certain companies, participants. And there is also an international from UK who have participated. And this is an international event this time. Till last year, we only had international professors giving you a talk. This time you're taken it one step ahead. So our dream is always to make it international. So from national workshop, you're going to make it international. I hope it will be done next year. Okay, not taking much of your time. Thank you, students. And finally, I want to wish our dean, thanks to our dean for helping us throughout, sir. There was, and then HOD, always she will tell, ma'am, come, we both will go and meet dean. So the work is done there and then. Thanks a lot, ma'am. And our chief guest, he never said, I got introduced here to Dr. Gopalakrishna through Dr. Sudesh. Then I said, let us try. Then he told me that, okay, he'll be back. You're back from Mumbai, right? So then we said, okay, I hope he'll come back here. Then he's here today. He came by Shatabdi and then straight here without having a coffee in the guest house. Thank you, sir, for your time. And as promised, I think we will have many students coming to you for internships. We had a time, we had some discussion during the coffee, and also his daughter has been here. So he knows what happens and how the internship and how our students, such beautiful and intelligent students we have at VIT, right? And Dr. Badrinath, yes, he is a VITian. He is a biomedical genetics, MSc and a PhD student. Yes, he's my student still, I say. But then he's grown a lot in a service, right? And I think he's the first person in biomedical genetics to enter a government job. So that was the main thing. I wanted him to come over here so that all are aware that, oh, life sciences, it can go till that level, right? So that was my main thing, so that everybody knows that there is some opportunity, and he was talking to a few of them also. And not but the least, students, thank you very much. Participants, thanks a lot for making this day. I promised Dean sir at 2:35 we will stop it. So I'm going to stop this. Thanks a lot. And we will all meet. We are going to have one more day going on tomorrow. Thank you.
Thank you, ma'am. And I hope DNA Days goes on and grows bigger each year. Now, I'd like to call upon Dean on the dais and felicitate our guest of honor and our chief guest. Now I'd like the Dean to give the memento to the chief guest, Dr. Gopal Krishna Ramaswamy. Now I'd like to introduce Dr. ARS Badrinath, who is a medical geneticist and district consultant of District Medical and Health Office, Andhra Pradesh. He is our very own alumni of Vellore Institute, and he pursued his PhD under the guidance of Dr. Radha Saraswati. He has been as a professor in Department of Transplant Immunology and Molecular Genetics, and also been nominated for Indo-UK Young Scientist Award at Chennai. I would like to call you upon stage and please share a few words.
Very good afternoon to all of you. First of all, I would like to thank VIT management, Chancellor sir for giving this opportunity, and also SVST for inviting me. So this DNA Day is a students event. As far as I concerned, ma'am, DNA Day is a students event, right? So I still remember the first day how it started, and still it is rolling because of the students. Acknowledgement is totally for students. Okay. So whenever I attend any of the chancellor's function in VIT, sir used to say that how VIT started, like how the proposal went to government, how MGR sir gave a single word of opportunity, and VIT started that time. And this day, it is giving education to thousands of students, and it is giving opportunity to grow. Opportunity is given, and we have to utilize. Okay. So whatever you are learning in every step, not only in classes, organizing like this events will ultimately benefit you in career. Okay. So coming to SBST, it is like a tree which will facilitates, which will into students like us, especially biosciences and BMG students. A dean, our sir, Siva sir, and the previous deans never said no to any event what we organize. We would like to thankful for this opportunity they have given to us. And also in coming years, the students has to organize very well, and it should continue. Okay. Okay. Yes, to all the students, hats off. Thank you.
Thank you, sir. Now I'd like to call upon the Dean, Dr. R. Siva, to give us a presidential address.
Thank you. The convener, Professor Radha Saraswati, the chief guest of the day, Dr. Gopalakrishna Ramaswamy, the guest of honor and our own alumni, Dr. Badrinath, Professor Asha Devi, head of the department, faculty colleagues, students, friends, ladies and gentlemen. Good afternoon to everyone. It is always a pleasure to stand before you, especially events like this. The DNA Day is one of the milestone, or I can say one of the events in our calendar of School of Biosciences and Technology. I'm happy that this is the 9th edition of it. As it was pointed out, April 25th, 1953, one-page write-up which was published in Nature. If I am right, I think it is 117 volume, page number 712 or something, I remember. So just one page write-up changed the entire scenario of the biological system. As well aware, being a biological students, DNA is the base for everything. And that is the milestone for understanding the structure of the DNA, by which, so many branches that it be, that has been grown in various fields. And without that, none of the research can be accomplished. So the DNA to RNA, RNA to protein, and so on. So I'm not here for a lengthy talk. We have the experts to discuss about that. But very interesting is that, as it was pointed out by the previous speakers, as well as accepted by myself, that the students are the backbone for conducting of such a big event. I'm really happy, and I also would like to involve in future as well. In fact, your juniors will come soon, and you have to inculcate that kind of the attitude to your juniors. These kind of the events should continue forever as well. And a small submit or request to the DNA Day, DNA team, is that we should also can have a broad theme, because this time it is very good that gene and metabolism, which is one of the important theme. Maybe we can also plan to have a diversified field of the DNA, because DNA is common for microbes, plants, viral, the human being, whatnot. So let us have the different session that related to microbial DNA, or plant DNA, or animal DNA, rather more focusing on the human. We can also have 1 or 2 events that will also cover all students of SBST to be part of it. Let the biomedical genetics lead the team, but the topics can be overall to be covered for all the students and participants. That will always, enrich our knowledge. I'm very happy to note that there are 272 participants are part of this event. Many of them online, and few of them I could see in front of me. And I'm really happy indeed. And I thank you for, making this event happen in a very nice way. I wish the very best, and congratulations to convener and the entire team of the biomedical sciences, and all the very best. Thanks a lot.
Thank you so much, sir, for your kind words. Now, I'd like to introduce our chief guest, Dr. Gopal Krishna Ramaswamy, CEO, founder, Therapeutics Innovation, Bengaluru. His specialization is in the field of microRNAs, who went on to complete his postdoctoral research in Yale University. He was formerly also associated with companies like Invitrogen, Thermo Fisher, and Genotypic Technology. Therapeutics, his brainchild, was the first company to get both Encounter Sprint and GeoMX DSP platform in India. Now, I'd like to call Dr. Gopal Krishna Ramaswamy to come and give us a few words.
First of all, I would like to thank Dr. Saraswati, and then Dr. Shiva, and then Badrinath, and then HOD ma'am. So it's a great pleasure to be here in front of all of you guys. See, I know, in fact, I had never thought I'll be giving a talk in DNA Day. I will tell you. The reason is I always loved RNA than DNA. So we used to go to canteen, myself and my friend, because since we love coffee, we used to say that, hey, do you want RNA or DNA? So RNA is coffee, and then the DNA is tea. So we thought DNA is more kind of we don't like. So it's just the French, how we used to talk about the DNA and RNA. So that's how our undergrad started. So it's I think one thing as a students, I think the teachers will teach their best. I know that, but sometimes the curiosity in you should lead to wherever you are going to go. So it's because sometimes the teachers are teaching for everyone, not for individual. You guys have to understand that your needs are sometimes different. You may have to spend more time than what they teach. It's not only this course, because whatever you learn is going to be a kind of a lifelong process. It's one thing I have understood here is, or anywhere, even I had fantastic teachers.
but when we asked so many questions to them, at one point they used to say, please go and then look for somewhere else. So they used to, I remember as an undergrad student, from GKVK, I went to IASC to ask a professor or a postdoc there. She was working. We wanted to know how the DNA structure is. Okay, it's around 1994 or 93, around that time. So we asked so many questions to genetics lecture. He said, no, I can't answer everything. Why don't you look at IAC? They do a lot of DNA work. Why don't you go and then check with them? So we went and then asked, like, that I remember her name is Aarti. Probably you might be Sharkad's wife. Right? Sharkad Malikarjun, you know? Yeah. Okay. I think they're diverse now, but so Aarti is the person who showed us the DNA gel and saying that we were kind of disappointed. But we thought DNA is like that, DNA is like this, and then they are showing a gel, and this is what do you make out of that? So it's like for every DNA looks same, isn't it? In a gel, if you see that. We were thinking of in chromosome how it will be, all those things, how the so it's a kindle which happens, ignition which happens, but ultimately what it takes, where it takes, you don't know. So it's some of these events still we remember. Whenever I go to IISc, I go now to sell certain things in IISc or to collaborate with certain people, but still I think that one moment which I went and then look for the DNA in the place. So again, so why I'm doing whatever I'm doing, it's always a question, isn't it? So I don't know how many of you have read George Bernard Shaw's principles or the quotes. One of the best quote I have heard is, everybody looks at the things and then ask why. I dream at things and ask why not. So it's a quote which I keep remembering myself and then asking, okay, it's something different you have to do. It's not something everyone is following, and then India needs people who are going to be different. We know that Watson looked at the structure and then we got the things. Why not the future Watson should be here, sitting one of you? So it is very, very important. All of you should think you are beyond anyone. So you think sometimes you are like Elon Musk, you are like Steve Jobs. All of you should keep thinking in that direction. Otherwise, it will be kind of we are always the followers rather than the leaders. All of you should be leaders rather than the followers. So that's one thing I always think people should be in that direction. So I think the last thing is there is a kind of a limit for what we are doing. So in the sense, there is no limit. You have to imagine, and then only you can innovate. So people should think of something beyond the things. So look at the cells and then what it can do or what it can't be done. So I heard recently, Sudesh was mentioning, there was some eye vision thing to solve the problem of the ophthalmologist. They created certain material. It could be some simple thing can solve quite a bit of problems. So whatever I showed is see, I'm quite old now. I can't probably invent that kind of the material. Always when you are younger age, there is no limit. I'll tell you, when I was a student, I used to think I will win a Nobel Prize. Okay, but only due course of time you realize, okay, that's not so easy. It's something beyond the thing. But presently the world is in such a way that India is in such a situation. Maybe 70, 80 years, or 30 years before what US was, and then how the energy was. That's what the India energy is. I wish all of you a great success. And then and another thing I was thinking, why not RNA day? I don't even know why not, isn't it? So it's after the first time I Googled the DNA day when I accepted the talk, I will be giving a talk. Then they said April 25th. So I'm doing it now. What is the reason? That's another thing I was thinking. Then he said there must be some postponement, all those things would have happened. So I don't know, maybe our company should start RNA day. Sure. Yeah, thanks. Good luck, everyone. Thanks for giving me opportunity and
Thank you, sir, for your kind words and for many confused young minds who will look up to you as an inspiration. And like you mentioned, to look outside the box and to become someone. Thank you. Now I'd like to call upon Dr. Asha Devi, our HOD, to give the vote of thanks.
So, good afternoon, everyone. I would like to take this opportunity to thank our Honorable Chancellor, Vice Presidents, Vice Chancellor, Pro Vice Chancellor, and the VIT management for their constant support and motivation in organizing the 9th DNA Day on Genes and Metabolism. I would like to thank our Chief Guest of the Day, Dr. Gopalakrishna Ramaswamy, Guest of Honor, Dr. Badrinath, keynote speakers, resource persons, and judges for accepting our invitation to be a part of this event. Thank you, sir. Thank you, Badri. My heartfelt thanks to Dean of SBST, Dr. R. Shiva, for his encouragement and support. Thank you, sir. I extend my gratefulness to the Associate Dean, Dr. Jayanti, Organizing Team Convener, Professor Radha Saraswati, Organizing Secretaries, Head of Departments, Event Moderators, and Student Coordinators who have been as a backbone in this program. I'm also thankful to the CTS, VIT, and the estate team members, always being there for any technical assistance. Thank you, sir. I would like to extend my gratitude to the staff members of SBST for rendering their support. Last but not the least, I would like to thank the participants without whom the event is not possible. Thank you again. Thank you all.
Thank you, everybody. Thank you, sir. Thank you, ma'am.
Students can assemble third, fourth.
Can everybody assemble in the middle for a group photo, please?
Wait, another one more.
Another photo. Oh. Another photo, I'll tell everybody.
Yeah, please inform.
Can you all please stay back? We need to take another photo. I'd request all the treasure hunt participants to please stay back and also the volunteers for treasure hunt to stay back. Stay back. Done? Okay, I think you can disperse. Someone told me. Yeah. We can disperse and only the treasure hunt participants and volunteers please stay back. हम लोग भी Wait. Yes. Everybody please stand for the photograph game.
Fast, you want to take.
All the treasure hunt participants, please stay back. The remaining of you, if you could disperse, please. Ready? Participants जिसने treasure hunt में लिया है इधर ही रुको. I told इधर stay back तो 2 बार नहीं इधर मतलब वो occupy करो जो भी बैठ. Yeah. Can all the treasure hunt participants please gather in the middle benches?
Gallery board.
It will happen in gallery itself. Start at 3. When's the next guest lecture? 3:30. Also, one more information, the next guest lecture starts at 3:30. It will be online, right? 3:30 at gallery itself.
Make an announcement. Treasure hunt will happen in gallery only. People are leaving in assembly.
Treasure hunt will happen in gallery right now. I announced, I told them to stay back. All treasure hunt participants, please stay back. The treasure hunt round 1 is going to happen in this room itself. If you could please inform your friends and call them back to the gallery. At 3:30 I mentioned, I informed. There are apparently 8 teams. Where? I told the remaining leave. How many times do I tell the same thing? Only the participants. Registered treasure hunt नीचे निकल गए ऑलरेडी वन many so many डीन का नाम मुझे पहले लेना था आई टू काफी एचडी एंड डीन का नाम याद नहीं आ रहा एंड इधर ना वो मोमेंटो भी दे रहे थे रे मुझे नहीं दिखा किसको दिया पहले एंड देयर लाइक and I'm like what do you mean
The second one when Sakshi indicated you.
I don't know. I thought they're handing over. Nee, I thought they're handing over both. So I didn't ask it separate. Something has it now. Check. I'll be calling out the team names. If you could just please raise your hands. Team 1, Bolepali Dhatri, Kirtana and Shrija. She's coming. And team 2 is Anushka, Takshita and Sudeepta. Team 3 is Subradyuti. She's not taking part. Harsh Pipalia and Rashmi Raha. He's alone. Next is Malin, Vishnu, Jyoti, and Mani Shankar. Then Munisha and Kalai Salvi. Okay. Sri Suganya, Harsha Preeta, Goki. Okay. Last team. And Sudeshna and Swastika. R3, Viswas. Volunteers who are not in treasure hunt, please sit in the last row and don't make any noise, okay? And one last team, Joshua Sequera and Somdatta Pal. Participants, another few minutes. We just want the other teams to just join us. All the volunteers, do you have a pen in hand? Okay, everyone settle? Okay, all the volunteers have been given the sheet. Once the time starts, and it's 10 minutes, once the time starts, they'll hand it over. Please write your team name on the sheet and team number as well. Okay, team number. You'll be given 10 minutes. Please do not ask the other teams for answers. It's a very easy treasure hunt level, and it's just logical reasoning. And okay, we're about to start. And no, please don't use phones. Anybody seen using phone or talking to other team members will be immediately disqualified. Thank you. Okay. Everybody submitting as early as possible will, according to the fastest first, whoever submits first will be qualified for the next round and also will be getting extra points. Okay. Please, the ones who are not part of treasure hunt, can you please maintain silence? Okay. Everyone ready? Volunteers? Okay, start.
Hello, is anyone there? Hello?
Hello, sir.
Hi, who's this?
Hello, sir.
Hello? Can you hear me?
Sir, we are extremely sorry that we will be conducting the session a tad bit late, sir. A tad bit later than what we expected.
At what time? About 20 minutes later?
Hello, sir.
Yeah. I see the message. Can you hear me? Hello? Hello?
We have 6 more minutes left.
Hello, sir.
Hello.
Yes, sir. I'm Vasudev, sir. I'll be hosting the session, sir.
Okay.
Sir, we are running late by a few minutes. So, would it be okay if we mute the session for a while?
No problem. Yeah, we can. I can wait. Just let me know.
Sir, would it be too much if I ask? Could you please stop sharing the screen? Will it be too much if I ask?
Okay, no problem. No problem. I'll stop here.
Okay. Okay. Thank you, sir. Thank you very much, sir. Thank you.
3 more minutes. Teams, hurry up. Last 48 seconds. Last 5 seconds. Volunteers, please be ready. Oh, time's up. Time's up. Put your pants down. All volunteers, please make sure you write your name on the sheet as well. All volunteers, put your names down on the sheets. Yeah, I hope the team has written their names on the sheet. At least group number and one person's name. And also, I think phone numbers as well, please. Sorry. Phone numbers as well, so that we can contact you about the results. One of theirs. One in the team. Everybody, please stay back. We have our next guest lecture. My God. So sad, actually.
Hello. Hello. Huh? On.
If everyone could please settle down and get ready for the next guest lecture. If everybody could take a seat, please, so that we can start the lectures. Everybody, you could sit down wherever you are. Please settle down quickly. Can yeah. All of you to the sides, if you could please come in the middle, because this is in the view for the guest. As it's an online lecture, if you could have a seat in the middle, please. Occupy the middle rows.
Make it missing because I didn't see we did probably swearing so tell them not to give any part. Only after stop one. You can don't go very far.
Kindly please settle down so that we can start.
Here, last.
Kindly, all the students in the back, can you please come fill up the front seats? Neelmani, if you could, come front. Can we start?
Hello, sir. Hello, sir.
Hello.
So can we start?
Whenever you are ready, I'm ready. Can you hear me? Hello.
Excuse me, sir. Can you hear us?
Yes, I can hear you. Can you hear me? Hello. I'm here. Can you hear me? I think you have some problem.
Hello, sir.
Audio. Yes, I can hear you. Can you hear me?
Dr. Nalashima, we are all here. Are you able to hear?
Yes, I can hear you. Can you hear me?
Yes, yes. Can you see the students? We are not able to see you.
No,
No, we are seeing only the slide.
I have the video. Let me do one thing. Just wait.
Yeah, 1 second.
Let me share the whole thing maybe. How about now?
We can't see you, sir.
I have my video on.
All right, sir. Let us check in. Just a sec, sir. Just a second, sir.
I can.
So maybe it's just a technical glitch, sir. Yes, sir. Now we can see, sir.
Okay.
First of all, let me apologize you, sir, for all the delay, and I'm thanking you for sticking with us and being patient. Respected dignitaries, faculty members, students, and all my dear friends, good evening to all. And once again, I welcome you all to the 9th National DNA Day. I feel proud and excited to introduce you all to the long-awaited lecture about implications of the complete sequence of the human genome for medical science by one of our most eminent and our dear guest lecturer, Dr. Nalashima Palanisamy. His area of interest is not something which is we don't know about. It lies in the discovery of
characterization of gene fusion in cancer, and understand their role in carcinogenesis. Dr. Palanisamy has received a number of accolades, which include 32 awards and 20 principal grants. He has worked in various prestigious institutions such as Genome Institute of Singapore, University of Michigan, Henry Ford Health System. And currently, he is working as the associate professor in the prestigious Michigan State University. I once again proudly welcome Dr. Nallashivam Palanisamy to the 9th National DNA Day, and hereby I officially hand over the dais to you, sir. Thank you very much, sir.
Okay, thank you. Thank you very much. I hope you can hear me better now, right?
Yes, sir. Yes.
Okay, good. Okay, good afternoon, everyone. It is early morning here. It is 6 o'clock in Detroit, Michigan. It is nice to see you all from the other side of the globe. I hope you all enjoying the day. I assume that it is a busy and a hectic day for you all. So in the next 45 minutes or 45 to 50 minutes, what I would like to do, I'm going to share with you, because as you all know, the Human Genome Sequencing Project, it was just completed by the beginning of this year. So I'm going to share with you what are the implications. So now we have the complete sequence. So what next, right? So that will be the main focus of my discussion today. So let me begin with, I think on this important day, you guys are also celebrating the bicentennial birth anniversary of Gregor John Mendel. So as you all know, he is the founder of the modern science, what we all call the genetics, although he himself did not coin the name genetics, but he laid the foundation stone for this modern science, which all impacting our daily life. So he published his experimental work on plant hybridization in 1866. I'm sure you all learning about more about these details of these experiments in your class, so I don't have to go in detail, but at least I want to recognize the birth anniversary of Gregor Johann Mendel. So it was in 1866 he published this work. Then at the same time, almost within 2 years, Dr. Waldeyer, he recognized the dark staining bodies. He called it as chromosomes, because chromo means stain and soma means body, the dark staining objects in the cell. He named those structures as chromosomes. So these, at that time, they have no idea what is inside this chromosome, right? So they will be able to recognize these type of chromosomes, both in plant material and in animal cells, but they have no idea what exactly is inside the cell. So it's all happened in the year, in the 1944, I would say, almost like 80 years from the publication of Georg Mendel in the year 1994, because of this eminent work of these three famous scientists. They showed that DNA is the genetic material that is lying in the cell, that is particularly in the chromosome. So that is the DNA, because until then, people thought protein is the genetic material, but they showed elegantly in their experiments. I'm sure you're all familiar that these experiments, that you learned in your class. So they showed that DNA is the genetic material. Then from 1944, within like 9 years, Watson and Crick, they solved the structure of the DNA. What is the chemical structure of the DNA? Although Watson and Crick, they did not do or they did not prove categorically that this is the structure. What they did in that paper, I encourage everyone, please read this 2-page article about the molecular structure of the nucleic acid that is published by Watson and Crick. They just proposed the model. So they don't have any experimental evidence to show that this is the genetic material, because at that time there were actually different type of DNA model has been proposed. Like some says like triple helix, left-hand helix or right-hand helix, all those different model, the DNA model have been proposed by different groups. But I think Watson and Crick, they were very confident that the structure, one of the structure that they proposed is the correct structure that could support the other functions, because it's not only structure, because based on the structure, what are the downstream effects of the structure? So if you read this paper, there is a one small paragraph where they clearly show that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That is the key. Okay, it is not only just the chemical is not sitting there, it is making the copy of its own. So I think as you all know, these are the four major important functions of the DNA. Okay, the DNA has to replicate itself. When the cell divides, the DNA has to go into it has to duplicate itself so that it will distribute equal amount of the DNA material to the daughter cells. So the DNA has to replicate. So the self-replication, so as you all know, is called a semi-conservative mode of duplication, meaning that one strand is used as a template. On that, the second strand is synthesized. So based on the semi-conservative mode of duplication, the DNA makes its own copy. And it's not only it is making its copy, it carries all the essential information for life. So that is encoding all the information for proteins and other things. Then this DNA can undergo mutation, and it can undergo recombination during meiotic division. And also importantly, the gene expression. So these are all the four vital functions of the DNA that is possible only with the double helix that is proposed by Watson and Crick. So just to show it will be inspiring for the students. It so happened for me that I was able to meet with him, with Dr. Watson. He's still alive today. I hope you all get a chance. If you come to United States, you may be able to see him. But I had a chance to meet with him for more than an hour, talking about my research and other things. It was a blessing for me. It is a memorable day for me. So based on the structure, so now we know that the DNA is lying in the chromosomes. So as you all know, the chromosomes are the key material for all the living organism, whether it is a plant or animal or human beings. Each organism has a finite number of chromosomes. Here I'm showing the exact number of these human chromosomes. This is 46 chromosomes. So in fact, this is my own chromosome. When I was doing my research, I made my own chromosome. So there are 46 chromosomes. Now, these 46 chromosomes, as you all know, it is arranged into based on the size and the position of the centromere. So the largest chromosome is chromosome 1 first, and the smallest is chromosome number 22. Then the X and Y chromosomes are separate. So now the monumental task is to sequence the DNA in each chromosome, right? So if you take the one haploid, that means because the chromosomes are in pairs, if you take just the one chromosome from each pair, you have 23 chromosomes, that is a haploid genome. So the size of the haploid genome is about 3.2 billion base pairs. So on the right side, I show you the exact number of nucleotide bases in each chromosome. I'm sure you can find all this information in the internet. You don't have to take any notes. So I think I want to request all the students, you don't have to take notes busily. I can actually share the entire PowerPoint presentation later, so you can actually view at your leisure later. So don't kind of get distracted for taking notes or anything like that. Just pay attention to the presentation. So now you have this many number of bases for each chromosome, the largest one. And also if you look at the number of genes that is located in each chromosome, okay, I give the number as well. So now the sequence is completed. So the main kind of the leaders who was behind all this effort is Dr. Francis Collins and Craig Venter. I think Craig Venter, he did an entirely different approach for sequencing the genome, but he is not able to compete with the effort by the National Institute of Health in the US, led by Dr. Francis Collins. So these are the two leading figures who was actually behind the completion of the Human Genome Project, or Complete Human Genome Sequence. So as you all know, the first draft sequence was published in 2003. So when I say draft sequence means the entire sequence was not complete. There are so many gaps in the genome. So they are able to, I would say about 90% of the sequence was ready at that time in 2003. So from 1953 to 2003, within 50 years, they were able to get the first draft of the human genome sequence. So this is completely done by the effort of Dr. Francis Collins. And here again, I was able to meet with him when he visited Singapore. At that time, I was a group leader at the Genome Institute of Singapore. The next one, so now, after the first publication of the draft human genome sequence in the United States Congress on February 27, 2003, they actually passed a resolution, concurrent resolution in the US Congress to celebrate the April 25 as the DNA Day. Okay, so this is the actual the document, the concurrent resolution passed in the United States to celebrate April 25 as the DNA Day. So now you know from 1953 they proposed the structure. Then in 2001 there is two papers. They showed the draft sequence. And they officially announced in 2003 completion of the Human Genome Project with a lot of gaps and other things. But in the April of 2022, now we have a complete sequence. There is no gap at all. Okay, so 100% of the human genome sequence is ready now. So this is the science paper that is published. That was published on April 1st, 2022. That has the complete. Truly complete human genome sequence, okay, from the draft sequence to that. So now, so this is the paper, this is the science paper. So please take, if you get a chance, please read this paper. So they have the complete sequence of the human genome published on April 1st. So now the entire chromosome. So when you see on the top, there is a small graph that shows these are all the number of additional kind of nucleotide bases that is added from the draft sequence, okay? So almost like another chromosome worth of DNA was sequenced. So now there is no gap at all. Every region of all the 23 chromosomes are sequenced, okay? So the major kind of delay for the completing the sequence was mainly the sequence of the centromere region was not ready, okay? Because the centromere region, as you know, it contains a lot of repetitive sequence. And also the sequence is quite variable between different populations, okay? So I think initially for sequencing the human genome, they used DNA from only like 4 or 5 individuals. They pulled the DNA and they sequenced it. And they found actually there's so many variations of the centromere sequence from different populations. But I think in the early 2000s, the technology was not ready to sequence it. But now, because of the advent of the next generation sequencing technology and the technique to sequence long reads of DNA, it was possible that they are able to fill the gaps in the centromere region as well. Because the centromere region plays a critical role, because especially in chromosome 1, 9, 16, these heterochromatin regions are quite variable. And also in chromosome 21 and 22, they are contributing for several genetic disorders like chromosomal non-bijection and other problems. So getting the sequence of the centromere is important. So now that is also complete.
Researchers have
So I have a
just
small
reached an
video,
important
just please watch.
Excuse me, sir. We are not being able to listen to what that video says, sir. Hello, sir. Sir, the video is not audible, sir.
Oh, I see.
Yes, sir.
I don't know what's going on. Probably the audio is not.
Sir, there would be an option showing share audio.
I'm sorry about that. If there is an audio problem, don't worry about it. I will share the presentation so that you can listen later.
Okay, sir.
Are you able to hear me now?
Yes, sir. You are audible. Just the video is not audible.
Right, right. At least you are able to see the video, right?
Yes, sir.
Okay, but I will share the presentation so you can watch later. Okay.
Okay, sir.
So now we have the complete human genome sequence, right? What is next? What are we going to do with that, right? So I would say that completion of the Human Genome Project is just the end of a journey, but it marks the beginning of another journey to interpret the information encoded in the DNA. Because sequencing itself, it took almost more than like 70 years to complete it. But the first phase of the sequencing is complete. Now the second phase is about the interpretation, how the information is packed in the chromosome or in the DNA, and how we are going to interpret that. Okay, this is the second phase. So although we have the map, but we don't know how to fold the map now. Okay, so that they have to figure out how to do that. So the reason I'm saying that, how to interpret the entire DNA sequence, is because of the enormous complexity and the information that is packed within the 3 billion base pairs. So if you take the entire human genome, about the 3 billion base pairs, so now we clearly know that there are only about 20,000 genes in human, okay? So do you think that only the 20,000 genes is just enough to make such a complex organism like human, or for that matter, any animal? So it's a so complex system. So I think there is more to learn, because if you look at these 20,000 genes, they account for only about 1% or maximum 1 to 2% of the 3 billion base pairs. So what happens to the rest 98 or 99% of the human genome, right? So it is there for a reason. So I think until recently, people thought that these sequences are junk DNA. So it is not junk anymore, because the humans have evolved from millions and millions of years. So all this DNA is there for a reason. So just because we don't know, we cannot call it as a junk DNA. So if you look at that, there are so many like small interspersed nucleotide elements and segmental duplications and several intronic sequences. So all these constitute the entire human genome. So if you look at other than this 1% of the sequence that is coding for the protein coding gene, the rest of the genome is actually coding for what is called non-coding RNA. Okay, so there are so many non-coding RNAs that are identified in the human genome. So that is called microRNA, non-coding RNAs, link RNAs, antisense RNAs, okay, and antisense, sense, intronic, and the protein coding genes. So this is the major challenge now. We need to interpret how many of these kind of RNAs are produced, because this is not part of the DNA. From the DNA, these RNAs are transcribed during the transcription process. Okay, so the DNA itself, the enormous information is condensed or packed in the DNA material. So then slowly it is actually transcribed into different type of RNA. So coming to the non-coding RNA, since it constitutes more than 98 or 99% of the human genome, it is important to characterize how many of these non-coding RNAs are there in our human genome, because it is there both in animals and plants, and everywhere it is there. So students, please go to this website. This is called noncode.org. So in this website, you'll be able to find all different type of non-coding RNAs that are identified so far. Okay, you'll be able to do that. So the term, when we say that non-coding RNA means, non-coding means, when you say coding means, it is coding for a protein, meaning that an RNA can be transcribed and it can be translated into a protein. So that is, we call it as a coding RNA. But when we say non-coding RNA means, we thought that these RNAs are not coding for any protein. But it doesn't necessarily mean that they do not contain any information or have any function. So we have to understand more in depth what these RNAs are really doing in the cell. So in this more updated version of the non-code, there are about 644,000 non-coding RNAs are identified, both in plants and animals. In humans alone, there are 172,000 non-coding RNAs. So just imagine, we have only 20,000 protein coding genes, but there are 172,000 non-coding RNAs. So that is another monumental task to understand what these non-coding RNAs are really doing in our body. And even in the mouse also, they have identified more RNAs, and in the plant also. So I think you will get the complete information when you go to this database. It is periodically getting updated. So if anyone is doing any project on this, you'll be able to get a lot of information from this website. So basically, when I say non-coding RNA is an RNA molecule that is not translated into a protein. So that is our understanding up to some few years ago, but now the story is changing. It is not really true that some of the non-coding RNAs do make some protein, which I will show in the next slide. So there are so many other type of RNAs there, the transfer RNAs and the ribosomal RNAs. So please remember that the transfer RNAs and ribosomal RNAs, they are not coding for any protein, because these are all the DNA transcription and translational machinery. They facilitate when the DNA is transcribed into messenger RNA. Then when the messenger RNA is translated into protein, then these RNAs come into play. But there are several other microRNAs like small microRNAs, siRNAs. These RNAs do have important functions, although they are not coding for any protein, but they have important regulatory functions. So as I told you, there are about 170,000 non-coding RNAs in the human. So now, although we have this 170,000 non-coding RNAs, we don't know the exact function of these non-coding RNAs. Although we know most of the protein coding genes, we know the function, what exactly they do. but we don't know the function of this non-coding RNA. So this is the next major task for any human genome researchers to understand the function of this non-coding RNA. And more importantly, now there are evidence that is coming up clearly shows that these non-coding RNAs are thought to contribute several diseases, okay, like cancer, cardiovascular disease, Alzheimer's, Parkinson's disease, muscular dystrophy, and several other genetic diseases are associated with non-coding RNA. Not only the protein coding genes, the non-coding RNA is also associated with the disease as well. So talking about the non-coding RNA, this is the new emerging concept. Okay, I'm sure if you are maybe you're familiar, or if you're not familiar, please pay attention to this, the new concept called micropeptide. What is a peptide means? Peptide is a collection of amino acids, right? When you have 100 or 200 amino acids that makes a protein, that is called a peptide. So most of the protein coding genes, they carry about more than 200 to several thousand amino acids to make a protein. But these micropeptides are only about 100 or 150 amino acids long. They're not more than these 150 amino acids. So these micropeptides are coded by small open reading frames. Open reading frames means there is a start site that starts with the methionine, amino acid methionine, then there is a stop codon. Okay, so they have a perfect open reading frame. So in mammalian genome, they started understanding that there are so many micropeptides exists in our mammalian cells. And previously we thought that those are all non-coding RNAs, but in fact they are coding for RNA now. So they have important function. These micropeptides, they play an important role in homeostasis maintenance of the intracellular mechanism. So please read more about this micropeptide. So now see, understand the enormous complexity of the human genome. We have the 3 billion base pairs, but that is not the end of the story, right? We know 20,000 genes, 3 billion base pairs, but there is more to that. Now we have this 170,000 microRNAs, and there are several, we don't know the exact number of these micropeptides. I think the more work is being done. So that could be another several thousand, right? So now we beginning to understand that these non-coding RNAs also shows abnormal pattern in cancer. Okay, you will see that some non-coding RNAs are expressed only in cancer cell, but not in normal cells. And they play an important role in regulation of many protein coding genes. So we thought that these non-coding RNAs are junk, but they are not junk anymore. So they play an important role in regulating the physiological program in developmental and disease context. So in cancer, non-coding RNA have been identified as oncogenic drivers or tumor suppressors in every major cancer type, meaning that oncogene means they can cause cancer, and the tumor suppressor means they can suppress tumor, and when these non-coding RNAs are lost, then the tumor will grow. So just understand the enormous complexity of the human genome. So this is the number of long non-coding RNAs so far identified in different cancers. Okay, so there are cervical, gallbladder, larynx, and all these different type of cancer. And these are the total number of non-coding RNAs identified in each cancer type. Okay. So I gave you the publication link. You can read more about this in the paper. So I cannot go into the complete details of that. Then another class of RNA is called microRNA. Okay, so there are more than like 2,600 microRNAs have been discovered in humans. Okay, so here again, go to this database called mirbase.org. So you'll be able to find the complete structure, the exact location, from which chromosome, which region these microRNAs are located. You will find all this information in this database. So these microRNAs are very important, okay? They regulate most of the genes in our genome, and the microRNAs have linked to many human diseases as well, okay? Or being pursued as the clinical diagnostic and therapeutic target. So there are many diseases like cancer, metabolic disease, viral pathogenesis, are all linked to microRNA associated defects. And now we started using microRNA as a diagnostic marker, and it is possible that antisense microRNA reagents can be developed as a therapeutic target. Suppose if a microRNA is overexpressed and that is responsible for a disease, you can stop the function of that microRNA by using the anti-MERS. Okay. So I can talk for another 2 hours with just microRNA alone. So I'm sure you can read more about this microRNA. I think it is part of the curriculum as well. Then another class of RNA in the human genome. So I'm just showing you how enormous, how complex our human genome is. It is not 3 billion base pairs, not 20,000 genes, okay? There are so many non-coding RNAs and microRNAs, micropeptides. Now, we started recognizing there is another class of genes called pseudogenes. It was first identified in 1977 in this frog, Xenopus levis, okay? First, they showed that the pseudogenes are the dysfunctional relatives of known genes that have lost their protein coding ability or not expressed at all. When I say pseudogene means they are essentially the same copy of a protein coding gene out of these 20,000 genes, but for somehow during the evolutionary process, the genes have actually duplicated. So what is called is a process pseudogenes, meaning that when a messenger RNA is transcribed in a cell, what happens inside the cell, it will get spontaneously reverse transcribed to make a double stranded, because you know messenger RNA is a single stranded. When it is reverse transcribed, it will become double stranded. So now the double stranded DNA will get inserted into a different locations in the genome. Okay, so this is how the pseudogenes are created. So like that, there are about close to 11,000 pseudogenes are identified in the human alone. So that is another major complexity that we learned from the human genome. So now please just follow through the information. DNA, human genome, 46 chromosomes, 3 billion base pairs, 20,000 genes. So now we have 170,000 non-coding RNA and several thousand micropeptides and more than 3,000 microRNAs. Now we have another 11,000 pseudogenes. So all these information is packed in this 3 billion base pairs. All right. So now the next, so here in this diagram, I'm showing that the pseudogenes are expressed in a tissue-specific manner, because some pseudogenes are expressed in only one particular tissue, and some pseudogenes are expressed only in cancer tissue. Okay, so we started learning more. So even in my research, I have identified a pseudogene called KLKP1 is involved in a gene fusion. It is fused with another gene, and it is expressed only in prostate cancer, but not in the normal prostate. Okay, so this is a major kind of discovery, first ever gene fusion involving a pseudogene in the history of the human genome. So we published this paper recently. So when you have time, please read this paper as well. So now we go to the next level. So now we know the complete human genome. We know how many protein coding genes, how many non-coding RNAs, how many microRNAs, how many micropeptides, and how many pseudogenes. So now you know that every cell has this 46 chromosomes. It has the 3 billion base pair, right? But it is not necessary that all the genes are expressed in all the time in all the tissues, right? So I'm sure you understand that, because every tissue has only certain type of genes are expressed. So now the next task after the completion of the Human Genome Project, now people are beginning to understand what are the genes, because we know the DNA is there in all the cells, okay? So with the DNA, we cannot do much. So what is the information that is coming out of this DNA in each different type of organs and the cells? So this is the portal called the Genotype Tissue Expression Portal, called the GTEX portal. I encourage you to go to this portal to see what are the different type of genes that are expressed in each type of human tissue. So they have the liver, lung, intestine, you know, all these different, about 53 different organs in the human. Now they started sequencing. because we know the DNA sequence. So we don't have to sequence the DNA over and over in each cell. Now they are trying to sequence the RNA. What are the RNAs that are expressed? Out of these 20,000 genes, how many genes are expressed in one particular tissue, and how many non-coding RNAs are there, okay? Each tissue. So in this portal, they are updating regularly. You will find the tissue-specific expression of certain genes in each different organ. So now another complexity is, so the GTEx portal shows the gene expression in all normal tissues, but there will be fundamental difference. Even if you take the same organ, suppose if the organ has a cancer, but the gene expression pattern completely different from the normal tissue versus the cancer tissue. So there is another database called Cancer Genome Atlas. So here again, more than 89 different type of cancers. Tissues have been sequenced. And because here again, the DNA is the same. Okay, so now we are focused on the RNA. What are the different type of genes are expressed? Please go to this portal. You will find all the information in different type of tissues. So here I'm showing one example. Okay, so this is the GTEx portal. There are about 53 different types of normal human tissue at the bottom. On the x-axis, you will see it. Here there is a gene called KLKP1. It is expressed only in prostate tissue. Okay, so all the tissues, they have these 46 chromosomes, the 3 billion base pairs, and everything, but only this gene is expressed in the prostate tissue. The KLKP1 is expressed only in among all these 53 different tissues. It is expressed only in prostate. Okay, so this is how you will be able to understand the expression pattern of different genes. So I'm just showing you one example. When you have free time, instead of playing in Google, searching for some unwanted information, please go through this portal. Just, you can type any gene, you can type any keywords, you will be able to learn a lot from that. And if you are an MSc student, you can actually even do some interesting bioinformatics projects. Okay, if you need any help, please contact me. You can ask Professor Dr. Radha. You can get my email address. Please contact me. I'll be able to help you all guys. So then this is the Cancer Genome Atlas. It's called the CBio portal that I showed you before. Here, there are 89 different cancer types. We know the gene expression. Okay. So here again, just understand, we are not talking about DNA now. We are talking about only the RNA, because the DNA sequencing is done. We know only the 3 billion base pairs. But how the information, is transferred or transcribed in different type of tissue. So when you take the cancer, here I'm showing one example. You can go to this database. You can visualize this data with few clicks of the mouse. There are 89 different cancer types have been sequenced, and the information is ready to view. So for example, the PIK3CA is an important kinase gene that is undergoing mutation in all these different type of cancer. The highest mutation is in the endometrial cancer, and the less number in other type of cancer as well. It is the same gene mutated across different type of cancer. So this type of information you will be able to get from this. So now all these data are publicly available. The whole genome sequence is available. The normal tissue sequence is available, and the cancer sequence also available. So anyone is interested in bioinformatics, you can develop interesting projects with that. So now the next challenge is, okay, so just I'm trying to give a logical flow of the information. Okay, so we started with human genome, 3 billion base pairs, 20,000 genes, microRNAs, non-coding RNAs, pseudogenes, micropeptide, then normal tissue, and cancer tissue, right? Now, the next level of complexity comes. So when you talk about a tissue, in a given tissue, you will have multiple types of cells. All the cells are not the same. Okay, when you say, for example, when you take an intestine or a skin, okay, so they have so many, like epithelial cells, mesenchymal cells, then all the kind of the basal epithelial cells. There's so many different type of cells in each organ. So now I think the scientists are beginning to understand what is not only individual organ, because we showed the information that is transcribed in the normal tissues in the GTEx portal I showed you. there is a whole tissue. Now they are digging deeper in each tissue. What are the different type of cells and what are the genes expressed in each cell type? So this is called a single cell. So we will be able to understand the gene expression at this single cell resolution. So this is the major revolution in the technology right now. Everybody is focused on now. They are trying to understand what is the gene expression pattern in the single cell level. So there is another database. It's called the Table of Sapiens. Okay, please go to this paper, or you can go to this database. It's called the Table of Sapiens. So because this project is now started, okay, so they were able to get information from 500,000 cells from 24 different type of tissues and organs from 15 normal healthy individuals. Okay, so now they started seeing what are the different type of cells carry the different genes. Okay, so they have lung, trachea, bladder, colon, duodenum, ileum, aorta. These are the type of tissues they are trying to understand now. So the single cell resolution is the next revolution in the understanding of the impact or the implications of the human genome. So now we know that the 3 billion base pair, the DNA is there, but what is expressed, how the different RNAs, microRNAs or non-coding RNAs, what are the things that are expressed in each organ and in each cell. So this is the next major challenge. I think this will be more inspiring for the young students. If you want to pursue a career in scientific research, there is so much to do this, especially the bioinformatics people, you know. All right. So now the next complexity comes. So now I told you there are about 20,000 genes. They are transcribed from the DNA. So that is the real kind of mystery of the human genome. The information is very tightly packed in the DNA. Now, when the DNA is unwinding, you know, it's tightly packed. When it started unwinding, there is so much is coming out. Say, for example, here, if one gene, it has an intron, exon, or, you know, it has 1 to 4 exons in this example. Some genes have more than 10, 20 exons like that. So it is not always, when a gene is transcribed, it is not always transcribed. It is always transcribed with all the introns and exons. It makes the messenger RNA. But when the pre-mRNA, when it is processed, okay, what is called pre-mRNA processing, during that time, what happens? It removes all the introns by the splicing mechanism, right? Then it will join all the exons to make the messenger RNA. Then the messenger RNA goes to the cytoplasm. It gets translated into the protein. So what happens during the pre-mRNA processing? Different type of messenger RNAs can be produced from a single transcript. So from the original DNA, it transcribes the pre-mRNA with all the introns and exons. Then during the processing, what happens? It can skip 1 or 2 exons. So you may have a messenger RNA with only from exon 1, 3, and 4. Another mRNA can have only exon 1, 2, 3 like that. So from a single transcript, multiple called isoforms or the variant forms can be created. Okay. So like that, from the 20,000 genes, more than 70,000 proteins can be produced. See here how the information is packed in the DNA. So now you have 70,000 proteins. Then it is not only that, the next level of complexity comes. So when the protein is formed initially, then each protein undergoes several post-translational modifications. So initial process is called transcription, then translation. So now you have another step called post-translational modification. That means after the protein is produced, it undergoes several additional modifications like acetylation, phosphorylation, glycosylation. These are the different type of modifications that is happening for each protein. So they are called proteoforms. So in this way, there are about 1 million proteoforms have been identified in the human genome. So now you can imagine the enormous complexity of the human genome and how the information is packed in the human genome. Okay, so the proteoform is defined a form of protein derived from a given gene with a specific amino acid sequence and a localized post-translational modification. You can read more about this in this paper. Here I'm showing one example, okay, using the insulin gene. Say, for example, you have a messenger, the DNA, the gene. So from this gene, there are two types of mRNA can be produced, like I told you, because of differential splicing. You can have two isoforms, and each isoform can undergo different type of post-translational modification. Like, they have some variations in the single nucleotide polymorphism, some then glycosylation may be added and phosphorylation as well. So now in the proteoform, you have the isoform, the messenger RNA, then it is undergoing glycosylation and also it goes phosphorylation. So these are the post-translational modifications, because these are all essential for the functioning of the protein. So like that, we have 1 million. So just imagine 3 billion base pairs, 20,000 genes, 170,000 non-coding RNA, 10,000 pseudogenes, then more than 3,000 microRNAs. Now you have 70,000 isoforms, then you have 1 million proteoforms. All right. So I think this is enough for the enormous, just to show the complexity of the human genome and how the information is packed and how it is revealed. So now the challenge is we need to understand how these different genes are expressed in different organs and different cell types in each organ, and what are the different type of proteins. Because I think that in the future, within another 10, 20 years, we are going to see all the information. Every cell, we know exactly what is going on in each and every cell. So this is an example of how insulin is produced in our body. So initially, the insulin mRNA is transcribed, then it goes translation, then it goes oxidation and the signal peptide cleavage, and export to the Golgi complex, then protease cleavage, and it goes to as a functional insulin. It has to go through several modifications before the insulin really works in our body. It is not directly coming from the DNA itself. The DNA codes only the information for messenger RNA. When the messenger RNA is transcribed, then it gets translated into protein, then the protein undergoes several other modifications before the protein really exerts its function. Okay, so you have to understand all. I think when you are studying in your class for your exams, please understand this fundamental, in a concept level, you have to understand it. Then it will be easier for you. So now I showed you, we have the human genome sequence. We know how many RNAs, how many pseudogenes, how many microRNA, and what are the different type of organs, what are the different type of cell types and everything. So now the next challenge is, what is happening to the gene expression in a disease condition, right? So all we are talking about so far is in the normal cell. So now what happens in a disease condition? So that is the next challenge. Now there are more than several thousand genetic diseases, like more than 7,000 genetic diseases have been identified. And when I'm talking about disease means they do have some abnormal gene function. It can't be normal. So you have to compare with the normal cell from a disease cell. There are so many, almost every gene is involved in one type of disease or other disease. So there is no kind of particular set of genes are involved in all disease like that. So this is enormous complexity. Go through this database, you'll be able to understand all different type of genetic diseases. So when I say the genetic disorder means every human ailment has some basis in our genes. I would say, I think that's what even my professor, Marimuthu, when I was a student with him, he always say that even the road accident has some genetic basis, okay? So only a person, he has to be mentally somehow abnormal at that, just for the time he may lose his function, something like that. That's why he's causing an accident like that. Maybe an exaggerating, but we used to laugh at the time. But now, after seeing all these things, I think he's right. Like every ailment has some basis in our genes. Without the gene, nothing can happen. So like even for disease resistance, some people take, for example, COVID. Some people get severe COVID, some people get less COVID. Like even if some people, they exposed to COVID, they don't get symptoms like that. There are so many variations in our gene. So that here I want to emphasize that although we have these 3 billion base pairs in every humans, but the sequence, when you sequence each individual, there will be some differences between each individuals. Even it is known that even identical twins have some differences, okay? So let me go quickly. I think we started late. I think I have another like 10, 15 minutes or 10 minutes. How much time I have?
You have enough time, sir.
Okay.
We are eager to listen to you as much as we can, sir.
Okay, okay, thank you. Yeah. So any inherited or acquired medical condition caused by a change or mutation in an individual DNA, okay, because some of the ailments in the genes are inherited. That means from parents, the children are getting it. So that is a inherited condition. Then some mutations are acquired during the lifetime of an individual, like cancer and other things. They are not, some of the cancers are heritable, but most of them are the somatic mutation, what we call, that is happening during the lifetime of an individual. So now the Human Genome Project and other genomic research really help to understand the multiple genetic factors acting together with the environment. As you know that every disease, there are certain diseases we know that definitely caused by a gene, but there are other diseases like cardiovascular disease and other cancer and other things. There is an interplay between both genetic and environmental factors. They have to act together. So you may have a normal DNA. Say, for example, if you are exposed frequently to radiation, like the people in Hiroshima and Nagasaki or the Chernobyl disaster, all the individuals were healthy. They have normal DNA, but because of this radiation disaster, they are exposed to the external radiation, then the DNA gets mutated, then they get all the disease. So this is the genetic and environmental factors, okay? So now the active research is going on, try to understand. So now we have the ability to understand the gene expression at the single cell level, right? So we have the power to see the differential expression pattern in different cell types. So now I think more research will tell that how we can improve the diagnostics and also develop some new drugs like that. So with that, so now the new era of the medicine is coming, what is called precision medicine. So like I told you before, every individual is different. You know, although we have each individual have 3 billion base pairs, but the sequence are different in each individual. Like a single nucleotide polymorphs and even a single gene, there may be small variations at the nucleotide base level that confers some resistance to disease, or it makes some people susceptible for certain disease. Say, for example, there is a gene called a CCR5. Yeah, there is a single base pair. There is a polymorphism in one base pair. If you have that, that polymorphism, those individuals are resistant to get AIDS disease. So even if they drink the AIDS virus, they never get AIDS because of the single base pair change in that gene CCR5, because that gene is important for allowing the AIDS virus to enter inside the cell. So if that polymorphism is not there, then the people are exposed. Okay, so because when you have this polymorphism, means that it's expressing an abnormal protein that gives some protection as well. So like that, we need to know, even if you know the gene, a particular gene is responsible for a particular disease condition, it may not be the same. All the individuals cannot be treated the same way. Okay, so first you need to understand what is the exact sequence in each person. Then you can decide based on what type of treatment they can offer. This is called precision medicine or personalized medicine, okay? So now once you know what is the genetic basis for each disease, now you can sequence in each and every individual. So you cannot generalize, okay, this person, I treated this person with this condition with this drug, I'm going to give the same drug because this other person having the same disease. So it may not work for the other person. So you need to know what is his gene structure or the gene sequence. So based on that, you can make a treatment option. It's called precision medicine. So you have to do either sequencing or PCR, and all different type of methods are available to sequence or to understand what type of abnormal protein is expressed. So now for the same disease condition, okay, so for the same disease, you can classify the patients into different subtypes based on the mutations or the aberrations they are carrying it. So this is called kind of personalized medicine, so you can separate the patient to different groups within the disease. Then if the drugs are available, you can give the right drug because each person may have a different mutation within the same disease, and you can select the appropriate drug for helping the patient. So here again, here it comes. When you are talking about the diagnosis of genetic diseases, so the markers can be at different level. It could be at the DNA level or at the RNA level or protein level or at the metabolite level. I think the theme of your DNA Day celebrations in the metabolism and genes. So I will touch upon a little bit about the metabolomics as well. So these are the different biomarkers that you can identify for different type of disease. Okay, if a DNA is mutated, that is a biomarker. If a particular RNA is overexpressed or a particular splice variant is overexpressed, so that is a biomarker, RNA-based biomarker. So then the protein, if the protein undergoes different modifications, where some protein is methylated in some individuals, in some individuals it's not methylated, so like that. So you can have some protein-based biomarkers. Then if there is a defect in the metabolic pathway, you will see some accumulation of particular metabolites, like phenylketonuria, alkaptonuria, and all those type of disease. One particular gene mutation. So if it is in a pathway, if one gene is mutated, so the metabolites start accumulating. From that level, it cannot be processed further. So these are all the different kind of biomarkers that you can identify for each type of disease, both the DNA, RNA, protein, and metabolomics level. So this is what I'm trying to say here. When a person comes with the disease, now I think this is a standard practice in the clinic. You take the sample biopsy tissue and you do the sequencing and understand what are the kind of genes that are responsible for that disease. Then you classify whether it is a mutation or a abnormal protein or a RNA, whatever it is. Then if the drug available, provide the drug, the patient may get treated, but sometimes the patient may not work. So then you have to do another biopsy to do more in-depth analysis to identify the right cause for that. So this is the type of approach that people are taking in the precision medicine or the personalized medicine now. So this is an example. Like in lung cancer, we identified there are so many mutations. Not all lung cancer patients are not the same. So they have lung cancer, but all the lung cancer patients are not the same. Okay, so there are at least 13 different types of mutations have been identified in lung cancer. So more than 50% of the lung cancer, we don't know what is the mutation because each mutation present only in a small subset of the patients. Okay, the KRAS is in a larger percentage, EGFR, but when you take the other mutations, they identify only a small percentage. So that's why now a patient goes to the clinic, they take a biopsy, they do the screening for all the mutations to identify which mutation is there. Then they will select that particular drug available for that particular mutant protein. Then the patient will get the benefit. So you cannot treat all the patients in the same way. So this is the essence of the personalized medicine. So there are about 359 genes, I think, have been identified in cancer-associated genes, but there are so many aberrations that are identified in cancer. Look at amplification, chromosome copy number changes, frameshift mutation, germline mutation, large deletion. These are the so many number of kind of defects that are identified in cancer. And there are a number of gene fusions, meaning that there are two genes located in two different locations in the genome. They break and they come and join together. This is called gene fusion. That happens only in cancer cells, but not in normal cells. So there are more than 69,000 genes, fusion genes have been identified in cancer for different type of cancer, So these are all, just to show you the enormous complexity and how much information that we are learning. So this is another therapy for lung cancer. There are different drugs that are available. So I showed you these mutations. So these are the name of different drugs that are specific for each mutation. Okay, so there's a lot of work going on to identify new drugs so that the patient get benefited. And as you all know, recently there is a checkpoint inhibitor. One particular protein, the PD1, is overexpressed in cancer. So that actually blocks that cell from recognizing. Our immune system cannot recognize those cells. If the cells are overexpressed with PD1, our immune system cannot recognize that as a foreign cell, so that it allows the cancer cell to grow in the body. So it is the remarkable work done by these two individuals. They showed that the overexpression of the PD1 is responsible for cancer. So if you can inhibit the PD1, so then your body immune system can recognize that cancer cell as a foreign, then it will attack and kill the cancer cells. So this is called immunotherapy, and they got the Nobel Prize recently, right? So quickly, I will show you what is the current status of the cancer therapy. Because although if you identify a mutation in here I'm showing an example for a skin cancer, there is a mutation in the BRAF, just a single gene, single base pair change, okay? At the amino acid position 600, there is a mutation. So now there is a new drug that is identified, it's called PLX4032, that is targeting specifically this BRAF mutation. So when you give this drug to this patient, the cancer is completely cured. Okay, just a single base pair mutation, single drug, you can completely cure this cancer. But unfortunately, what happens after like 23 weeks, the cancer came back in this individual. It is because, like I told you before, every cell in the cancer is not the same. So now you need to understand at this single cell level, what are the mutations in each type of cell. Because now what happens here is the majority of the cancer cells carry this mutation. So the drug will work only on the cells that carry the mutations, but not on the other cells that do not have the mutation. So once these mutant carrying cells are eliminated, the other cells started growing, and we don't know what exactly is the mutation in the other type of cells. So now, like I told you, we have to do the repeat biopsy, then you have to do the more in-depth analysis to identify any new mutations to help these patients. If there is no new mutations, if there is no new drug, unfortunately, we cannot do any help for this individual. So this is the current status of this cancer therapy. I think that's where the single cell analysis is coming into play. Okay, so this is the remarkable kind of development in cancer research and also in the genome research. Analysis of single cells, understand the gene expression pattern at the single cell level is the new development or the new focus in cancer research or any genetic disease. Okay.
Excuse me.
All right. Just quickly, just a few more slides. I will finish this. Talking about the metabolism, I think I don't have much time. I don't want to go into the details. I'm sure you all studying in detail more about this. But just a few examples. I think from 18600 itself, the first inborn errors of metabolism was recognized. It's called alkaptonuria. That was the disease. It was called the black urine disease. So this is a rare autosomal recessive disorder because of the defect in the processing of the amino acid tyrosine and phenylalanine. These individuals, they accumulate certain metabolites, and they have this severe disease. Like that, there are many other several metabolic diseases have been recognized. So now there is another new field is coming up, is called metabolomics. From genomics, now they have a metabolomics. They are trying to characterize different type of metabolites. What are the different metabolites that are actually processed in each type of cell? So that is another active area of research is going on. So this is an important paper that is published recently based on the genes that are involved in high metabolism and low metabolism. Okay, so please read this paper when you have the chance. So they were able to identify that these are the different altered metabolomic genes called the most altered metabolic genes. Look at the figure B. So these are the different genes involved in metabolism, and they are actually affecting more all different type of cancers, like metabolism of lipids, metabolism of carbohydrates, metabolism of amino acids and derivatives, and the cofactors and the biological oxidations. So these are all the different metabolic processes that are affected in cancer cells, and they are responsible for cancer. Okay, so this is another big area of research going on. So please read this paper. This is an important paper that came out recently. So these are all, yeah, go ahead.
I'm extremely sorry to interrupt, sir. I just came to know that we have to leave the auditorium by 5. So we move on to the Q&A session as early as possible. If it's
Okay, I will just finish with the, I think this is the last, anyway, I'm going to share this slide. You guys can read this, okay? There are a lot of breakthroughs are coming up in the genomic research. And the last but the least, this is the big effort that is being conducted in the US. They are trying to sequence 1 million people now. Okay, so now we have the sequence, but they are trying to sequence about 1 million healthy individuals. Initially, they want to get the sequence done. Then they are going to follow up these 1 million individuals during their lifetime to see, because they know what are the genetic variants that are present in each individuals, and they will follow up about the life conditions, food, lifestyle, and everything, and they will follow up what are the diseases that they are developing, and they are trying to connect their genome with the disease like that. So this is a major effort that is going on. And if you have time, please go through this, some of the bold predictions for the Human Genome Project by 2030. What are the new things that are going to come up? You can read that, okay? So the last but not the least, so I want to say that the DNA is precious material. You know, you may have $1 million or whatever it is, but you may not be able to get food, right? You know, just imagine Steve Jobs, the founder of Apple. He had billions of dollars, but he is not able to help himself because he suffered from cancer called pancreatic cancer, and he died at the age of like 56 or 57. So my take-home message now for the young students, you are young and healthy now, your responsibility is protect your DNA, okay? Your DNA is the precious wealth that you can have. Until your death, if you have good, healthy DNA, and your life will be more happy, and you can earn anything you want in your life. So it's important that just to avoid these things in your life, okay? Tobacco, alcohol, and also you have to limit calories. Don't eat all the junk food, whatever that is coming in your way. Eat healthy, and you have to avoid cancer viruses, and just consume a lot of fruits and vegetables. And these are all the important key for preventing cancer and also protecting your DNA. Your DNA is your wealth, okay? I think that is my important take-home message on this important DNA Day. I want to thank everyone for giving me this opportunity. I hope it is useful. Please feel free to ask any questions, or you can email me anytime. I'll be happy to answer any questions. Thank you.
Thank you, sir. Thank you very much. So
you started late. I think I was able to finish within 1 hour, right? Thank you. If you have any questions, if you can type the questions if you.
Could you please stop screen sharing, and can we see you in a larger screen?
Okay. Can you see me?
No, sir. No, sir.
Share screen.
So the Q&A session is open. If anybody has a doubt, can raise their hand.
I think you can see me in the gallery, right? Are you able to see?
Yes, sir. Yes, sir.
Okay.
Can see.
They say there is no audio. Can you hear me?
Yes, sir.
Okay.
Audible, sir.
I think everybody's tired after lunch and a hectic day, right? Please ask some questions.
I think more of a. Yes, sir.
Morning, sir. My name is Samboti Bhattacharya, and I'm from Integrated MSc course in Biotechnology. So my question to you is, you showed in one slide about the hot air or HOTAIR LNC RNA. Can you please explain the function of those?
I don't have the complete details, but these are some of the anecdotal, examples of this non-coding RNA. There are several other examples. There is another one called NEAT1. We showed the so most of this non-coding RNA, they function more like regulating the function of other protein coding genes. Okay, what we call as competing endogenous RNA. So what happens when the messenger RNA is produced, when it is transcribed, some of these non-coding RNAs are transcribed in the antisense orientation, okay? So the sequence is they have some sequence similarity at the 3' end of the protein coding gene. So this non-coding RNA, like the microRNA, they go and bind to the pre-messenger RNA that is transcribed for a protein coding gene, and they will block the function so that the messenger RNA cannot be translated into the protein. So they will suppress the function of some protein coding gene. So these are some of the known functions, but there are so many other things that we don't know exactly. So like when you take the microRNA, they bind to the 3' untranslated regions. So they will inhibit the transcription of some protein coding genes. Similarly, like the pseudogenes, I think there is a good example. There is a pseudogene for the PTEN, the tumor suppressor gene. So when the pseudogene is expressed, it is expressed in the antisense orientation. It blocks the translation of the protein coding gene. So this is how most of the non-coding RNA function is known so far, but there is so much more to learn from this.
Yes, thank you. So my next question would be, so you spoke about the long non-coding RNAs. So like what is the scope of spatial transcriptomics in the discovery or the function or discovering the function of long non-coding RNAs in the case of cancer?
Exactly. When you do the spatial transcriptomics, that is not understanding the function. When you do spatial transcriptomics, you will know what are the genes that are expressed differentially in different cell types. Only that much you know, right? So once you dissect out what are the different cell types expressing different transcripts, understanding the function is totally different, right? So you have to kind of clone this kind of RNAs and do several in vitro cancer biology or some biological experiments to understand the actual function. So what you need to do, you have to do some knockdown experiments, just to block the expression of this particular non-coding RNA, see what happens in the cell. So if a cell that do not have that particular non-coding RNA, try to overexpress this and see what are the changes that is happening. So I think, because I did not mention about the spatial transcriptomics, because I think I just touched upon the single cell analysis that is part of the spatial transcriptomics. So the spatial transcriptomics is a revolutionary technology. You can select a certain number of markers. You can analyze the entire tissue, all type of tissues that you can analyze. So that will give you the expression pattern in different type of cells. Only that much we know. So once you know what are the cells, what are the specific cell type that express that particular RNA, then suppose if it is expressed only in the immune marker, only in the macrophages like that, then you know that the function of this, because when you look at the organ as a whole, you know that RNA is expressed in that organ. But when you go to individual cell type, okay, this is the cell type that is expressing this particular RNA, then you target that. So this is exactly what happened when people are working on the COVID, right? When they look at the lung, they did the same type of single cell analysis. They looked at the ACE2 and the TMPRSS2 gene. These are the two key genes that is involved for the COVID virus entry and the processing. And now they are able to dissect out what is the specific lung cell type that is expressing this ACE2, okay? So what happened was they identified there is a particular stem cell in the lung, kind of stem cell, that is the cell expressing high level of ACE2. So that is the main reason, since the virus attacks the basic stem cell, then the lung that loses the regenerative capacity. So once the what is called the stem cell is gone, the lung, it cannot kind of regrow again. So that is the main reason most of the people, they suffer from lung infection and they succumb to death. So that is the key, yes, you asked the relevant question. Doing the kind of spatial transcriptomics, we gain a lot of knowledge that the differential expression pattern of the genes, then you can actually try to understand the function, how it is doing in each cell type.
Thank you very much, sir. Due to time constraint, I fear we may have to conclude the session, sir. We are extremely sorry. On behalf of SPST VIT Vellore, I thank Dr. Nalashivam Palanisamy from the bottom of my heart for taking time and effort from his tedious schedule. His presence and knowledge helped venerate this event to a great extent. Thank you, sir. Thank you so much for being with us and for being patient in the beginning also. And I thank each and every participant in this event and all the faculties and all the organizers who did this fantastic work of doing this. So I officially conclude this event. Thank you,
I
sir.
see Professor Radha is there. She has any word to say? Okay, thank you very much. Thanks everyone for attending. Please feel free to send any questions later to my email. Thank you.
Yes, sir. Thank you.
I'm going to leave now.
Yeah, yes, sir. You may leave, sir. Thank you. Just a sec.